Good afternoon, everybody. Welcome back to class. Hopefully you guys had a good and a productive spring break. Let's talk game plan. Our game plan is to finish today our lecture on the skeletal system. We have to talk a little bit more, go back and talk a little bit more about our skeletal muscle anatomy. And then finally we'll come back, and now that we've learned everything we ever wanted to know and more about skeletal muscle, we will then briefly talk about smooth muscle and cardiac muscle and discuss those and how they're similar and how they're different from what we've learned in skeletal muscle. The other thing that we need to talk about is the exams, because on Thursday you have the exams, and as I've mentioned, but I will mention it again here, the exams are available from 10 a.m. To 6 p.m. You need to complete the exams during that time, so make sure you plan accordingly. I am mostly done with your exams. There's a little bit of tinkering I need to do, and then I need to test it for any kind of problems or things along those lines. But in general, I have an understanding of what the exam is going to be like, so I wanted to talk a little bit about that. Your lab exam, I'll be honest, this is the most brutal exam I have ever written in my entire life. Not necessarily that it's hard for you guys, but it was hard for me to make, because basically when I go into an exam, normally I'm making one version of it. For these exams, they are basically unlimited versions. So basically what I had to make was a test bank that literally had every muscle and every action and every origin and every insertion and every structure. I did over 300 questions for this lab exam. All right, so again, every single thing that is on the plate you are responsible for. Now, that doesn't mean you have to answer 300, thankfully, but this is a big exam. There are going to be 80 questions on this exam, and so obviously it's going to be then therefore worth 80 points. 1 point per question. The format for this is going to be similar to what we had on the last exam. One question at a time. And you cannot go back. All right. So again, it is going to be one question at a time. Once you answer a question, it's locked in. You cannot go back to it. So you go through them first once. And again, this is similar to the format we had in the classroom. The only difference is we did get a brief review at the end. And of course, this is timed. You will have a total of 90 minutes to be able to answer all of those questions. So you'll have 90 minutes. You need to plan accordingly with your time when you're going to be doing that and how you're going to be doing that, so that, again, if you realize that you're on question 20, right, and you've already spent 70 minutes on it, then you definitely need to pick up the pace at that point. If you're on question 20 and you've been doing it for 5 minutes, then you can realize you can slow down a little bit. You're doing good time-wise. But so this one is going to be similar to the format that we had before. One question, one at a time, cannot go back, and it's going to be 90 minutes to answer those 80 questions. The lecture exam is going to be more like what we had in the classroom. It is going to be a total of 30 questions. Those 30 questions are going to be 20 multiple choice. There are going to be 3 fill in the blank. So let's actually do it this way so it all fits in. Multiple choice, 3 fill in the blank, and 7 essay questions for you to be able to complete for that. And again, here the format will be more like what we had in the classroom. The format for this is you will see all the questions at once. Oops. Oops, if I spelled together correctly. So when you open up the exam, You will see all 30 questions. First, the 20 multiple choice questions, then the 3 fill in the blank, and then all the essay. So if you want to look at the essay questions first, see what they are, and then think about them while you fill in the multiple choice, you have the chance to do that. You can bounce around however you want to within that, so that you have the opportunity to be able to do that. And then once you've answered all of the questions, then you submit the one exam as once. Are you able to open up the practice proctorio exam? No, unfortunately, because of the way the proctorio works, it isn't easy for me to do that. I can set up a, so if anybody was not able to take the practice exam before and fill the full proctorio, if you contact me, I can set up a quick, simple test for you to, a proctorio test to do, so to make sure that it is compatible with your system. So if you want to be able to do that, send me an email and I will set that up today. So you'll see all the questions together, and then you submit it as once. And then for this exam, the lecture exam, you will have a total of 2.5 hours, or what essentially is 150 minutes. One last thing for the lecture exam. I have given you a tool. That tool is a basic whiteboard. When you are taking your proctorio exam, on the left-hand side is a tab that says Tools. And in there, there is a basic whiteboard. So that whiteboard is something that you can bring up. You can do your own drawings if you want to do drawings. You can write notes on there. So there's no scratch paper or anything like that on this exam. But if you need to take notes or you want to take notes before you write these things down, you can draw, you can type, you can color, you can do those things. So there's a basic whiteboard tool for that that you'll have access to. And like I said, it's just on the left-hand side when you're taking the exam under the tools that will be available. That basic whiteboard is not available during the lab exam. So it's only for the lecture exam that you would need that. And I'm not sure you need it anyway, but I wanted to have it available. The other important point that I wanted to make sure that I made is to, when you are taking the exam, to make sure you scan your work area. I think literally no one who took the practice exam did this properly. I got plenty of pictures of the plants in people's back of the house. One person, it was in my 431 class, actually walked around their house during the scan of it. Obviously, don't do that. Take one station. I don't care about your ceiling fans or anything else. I want to see your work area to make sure that, again, you don't have your phone, you don't have any notes, you don't have any other materials that are there available. So make sure you point your camera down or bend your laptop down so that I can see your work area and see that, because that is more important than what's hiding in the corner of your room. Oh, great question. Forgot to mention that. Yes, the lecture exam also, again, like our lecture exam in the classroom, will be 100 points. So again, this will be, so it will be 100 points, just like the normal one in the classroom. So this, the format for these is very similar to the first 2 exams that we took. And again, we were rushed with that third one, so that one was kind of a bit of an anomaly. But this should be much more like what we had before. Like I said, there were over 300 possible lab exam questions that you will be given. Again, using models, using illustrations from your textbook, using illustrations from your atlas, microscopy views, again, bodybuilder pictures, all those things that we talked about before are still going to be on this. And you need to know your origins, your insertions, your actions, origins, insertions, actions, and obviously the names of the muscles, names of the structures, bones. As I mentioned, there are going to be bones on this exam as well. You need to be able to recognize attachment points and know what bones attach to them. So all those things we talked about are going to be on there. Same thing with the lecture exam as well. For the lecture exam, like I said, you're getting a random sample of 20 multiple choice questions out of, I think there's something like 77 total that I came up with. You're getting 3 fill in the blank out of, I think, a total of 20 that I came up with, and 7 essay questions out of a total of, 50 something like that. So you are going to be getting a random sample of questions that you are going to be responsible for. And like I said, in some ways, I have no idea what this is going to be like. Because in some ways, like I said, the lab exam is a great example. Literally every single possible answer I had to make a question for. If you think of the essay questions, literally every single possible essay question I could think of is in there because it's something that could possibly come up. So all those 7 essay questions that we talked about that I guarantee there's going to be 2 of, I guarantee there's going to be 2 of those. And there may be more. And again, it's random. So your questions may be a little bit harder, and maybe a little bit easier. Hopefully you'll get a random enough mix that everybody's will be about even. But we'll see. Haven't done this before. The one advantage I will have is my 431 class. They have their exam actually on Wednesday. So hopefully if there's any problems that they have, I can learn from that to help make ours more smooth on Thursday. But things went relatively well the last time we did this, so hopefully that will be the same here. The only difference is now we're using the Proctorio, so that may add a few more kinks than we had the first time we did this, but hopefully this format will be more straightforward. It's more similar to what we had on our first couple exams. And again, you see that in the points as well. All right, any questions on any of that? Dead silence. Excellent. All right. So either everybody's already bored and asleep, or that makes some semblance of sense. And so we can move on to the material at hand. Let me go ahead and save that whiteboard so that I can post that like we've been doing. And then with that, we can switch gears to go back into our lecture. All right. We left off last time basically talking about skeletal muscle, and for the most part thought we had learned everything we possibly could have learned about skeletal muscle, except for the fact that everything that I've said up to here has now been a huge bold-faced lie. All right, well, that isn't entirely true. But what is true is up to this point, we have been talking about skeletal muscle fibers or skeletal muscle cells, sorry, as if they are just one thing. Right. However, as we've learned before, cartilage is not just one thing. There are 3 specific types of cartilage: hyaline cartilage, elastic cartilage, and fibrocartilage. As we talked about, all cartilages share some characteristics, but there are also some significant differences between those cartilages. And it turns out skeletal muscle is the exact same thing. Skeletal muscle actually has 3 specific types. So there are 3, oops, so I spelled specific right, specific types of skeletal muscle. And so now that we've talked about skeletal muscle as a singular thing, we actually have to look at the skeletal muscle and see and distinguish those differences. Now, when it comes to these differences, there are some main ways that they are different from each other. The first is in the fiber diameter, right? And the fiber diameter, obviously, all of the muscle cells can vary in length, but what I mean by diameter is basically what the width of it is, right? When we talk about diameter, what I'm really talking about is the width. However, what makes a cell wider is basically having more myofilaments. Or let's say maybe more specific, myofibrils. And of course, if you have more myofibrils, well, that of course means that you have more myosin and actin. If you have more myosin and actin, you can get more power strokes. And if you get more power strokes, you produce more tension. So notice when we talk about fiber diameter, what we're also really talking about is the strength of the contraction. How powerful of a contraction can that muscle cell have? Because the more myofibrils that muscle cell has, the more power strokes, the more myosin, the more actin, the more tension it can produce. So one of the ways these things are going to be different is by basically how thick the cell is, how wide the cell is, and that also influences how strong that cell is. A second way these will differ from each other is in the speed of the contraction. Right? One of the things that we talked about is that on average, oops, wrong button. We said on average, a myosin head completes 5 contractile cycles per second. Right? That was what we said before, but the key word there is on average. What that means is that there are actually 2 different types of myosin heads: slow myosin heads and fast myosin heads. Not surprisingly, fast myosin heads go fast, produce far more than 5 contractile cycles per second, and slow myosin heads do less. So if you have slow myosin heads, your muscle cell contracts slower. If you have fast myosin heads, your muscle cell contracts faster. Another thing we talked about is the importance of myoglobin. Myoglobin, of course, remember, is that hemoglobin. I don't know why this is all yellow. I don't like the yellow. Let's switch to simple black. Is a hemoglobin-like protein in muscle cells. And like hemoglobin, it stores oxygen. Oxygen, of course, is what we use to completely break down glucose, right? And in that process, we called aerobic respiration. Right? And so we talked about how all muscle cells have a large amount of this myoglobin to store oxygen for that aerobic respiration. And it turns out that's not entirely true. Some muscle cells have a lot of myoglobin. Now, again, myoglobin, like hemoglobin, has an iron molecule. And that iron, oops, iron, right? Has iron, and that iron is used to bind the oxygen. And as we know, in our red blood cells, when oxygen and iron come together, they give us a red pigmentation. So notice cells, muscle cells that have a lot of myoglobin will have a reddish coloration to them. Cells that have only a little myoglobin in them, right, is going to be more whitish in color. And I know this idea of colored muscle doesn't make sense because all muscle is red, right? Well, that's not necessarily true, right? Right around the corner is going to be Thanksgiving. Okay, it's not really right around the corner, but eventually it's going to be around the corner. And hopefully at that point, families will be able to gather. And when families gather, what is the big question that everybody always asks you? Well, usually they're things like, when are you going to get a job? When are you going to get married? When are you going to have kids? When are you going to do all those kind of things? But the other big question that get asked on Thanksgiving is white meat or dark meat, right? We have those breasts, we have those wings that are the white meat, we have those legs, we have the thighs that are the dark meat, right? When they talk about white and dark meat, light and dark meat, what we're really talking about is red and white muscle fibers. These differences in the red and the white, the ones that have a lot of myoglobin or ones that only have a little bit of myoglobin. Muscle cells differ in their capillary supply. Muscle, for the most part, is well vascularized, but some muscle cells are going to have more blood coming to it than others. That's going to be useful because it's going to be able to bring oxygen more rapidly to the area and glucose more rapidly to the area. So there are some that are going to get more oxygen and some that are going to get less. Muscle cells are going to differ in how much mitochondria. Some are going to have a lot of mitochondria and some are going to only have a little bit of mitochondria. Right? If you think about it, if you're one of these red cells that has a lot of myoglobin, you've got a lot of oxygen. So if you have a lot of oxygen, you're going to want a lot of mitochondria, right? Because that oxygen is used to take that sugar into the mitochondria and break it down, completely. If you do not have a lot of myoglobin, you're not able to store a lot of oxygen. And if you're not able to store a lot of oxygen, there's not a lot of point in having mitochondria, right? It's the same thing with the capillary supplies. If we have a lot of capillaries, or strong, let's say it that way. I say a lot of capillary, large, I like that better. Large capillary supply to a blood vessel, it's going to be easier for it to get oxygen. More myoglobin is going to make it easier to store oxygen. And so it makes sense to have a lot of mitochondria. Conversely, if you have a small or scant blood supply, if you're not able to store a lot of oxygen inside of you, then there's not a big point in having mitochondria, because the mitochondria are only useful if you have oxygen. And so notice these three things kind of work together into one of the other big differences for our skeletal muscle cells, and that is how they produce ATP. If you have a lot of myoglobin, if you have a large capillary supply, if you have a lot of mitochondria, then it is easy for you to produce energy with oxygen. Whereas if you don't have a lot of myoglobin, if you have a scant capillary supply, if you have very few mitochondria, then you're primarily going to produce ATP, or energy. Let's be consistent. Let's spell it right. Without oxygen. Okay. And of course, as we know, anatomists love to name everything. So we actually have names for cells that produce ATP primarily with oxygen using aerobic respiration, or produce energy without oxygen using anaerobic respiration. And those are oxidative and glycolytic. So two primary ways of producing ATP, aerobically or anaerobically. If you're aerobic, we consider you to be oxidative. If you're anaerobic, we consider you to be glycolytic. Right? So these are the characteristics that differ in our skeletal muscle cells. Let's see some examples. Here is our first type of skeletal muscle cell. And again, it says muscle fibers, but again, remember, these are all skeletal muscle cells, again. And the first and the most common is a fast glycolytic fiber. This fast glycolytic fiber is large in diameter. And if it's large in diameter, what does that also mean about it? I'll wait this time for an answer just to make sure not everybody's asleep. The cell is large in diameter. What does that also mean about the cell? Absolutely, it produces more tension. So this one is going to produce powerful contractions. All right. There's also a lot in the name. This said, the name of this cell is a fast cell. In this case, by fast, what do we mean? Of course, we mean fast contraction. And what causes that fast contraction? Fast myosin heads. Excellent. Excellent. So it's fast in that it has those fast myosin heads. The second part of the name is that it's glycolytic. Glycolytic, of course, means, as we said, that it produces ATP primarily without oxygen. All right. So it is anaerobic. Being anaerobic, then, we would guess that there is less mitochondria to these cells, right? It's going to have more of a scant blood supply to it because it doesn't need to get oxygen as readily because it's not going to necessarily use it. And again, if it's not going to use oxygen, it's going to have small amounts of our myoglobin. Because of this, these cells are the cells that we consider, oh, I'm not going to be able to use white, so I'll use it here in yellow. These are our, what we call our quote unquote white muscle cells. Come on. There. Now, there is one more issue of this. Remember, if it's producing its ATP anaerobically, remember, how many ATP do you get from one glucose with that? One glucose produces how much ATP if we don't use oxygen? Give me a number. 2. Excellent, 2. So it's very inefficient. It's like having that Hummer. If you drive a Hummer, right, one of the things that Hummer has to have is a massive gas tank. It needs that massive gas tank because it is very fuel inefficient. If you had a 10-gallon tank, you wouldn't be able to get from gas station to gas station with your Hummer. So you need a massive, huge gas tank. And the same thing's true here. If these cells are going to be less effective, they have, while they don't have myoglobin, they don't have mitochondria, they need a ton of glycogen, right? It needs a huge gas supply. But even with that big gas tank, these cells are very inefficient. So while these cells can produce very powerful, very fast contractions, can those contractions be maintained for a prolonged period of time? No. And so these cells are ones that fatigue quickly. Right? So these are cells that help us to throw a punch or throw a ball or lift a weight, right? Fast, powerful contractions, but ones that cannot be sustained for a prolonged period of time. I've put my list here, but let's go ahead and erase it. And then we have the pretty pictures that do the same thing. These again are our quote unquote white cells. Very few mitochondria. They produce their ATP anaerobically. They need that large glycogen storage to them. They produce fast, powerful contractions, but they fatigue very easily. And like I said, this is going to be useful for intense movements like lifting a weight or throwing a ball. All right, questions on that one? All right, excellent. Let's talk about our second type of skeletal muscle fiber, and that is our slow oxidative fiber. Again, it's got erased, so I'll make sure we put it here to emphasize it. These are all skeletal muscle cells. Slow oxidative fiber, remember our fast glycolytic were the largest. Our slow oxidative are the smallest in diameter. And of course, small in diameter, of course, means that it is produces a weaker contraction because it does not have as many myosin and actin in it. Also, as we talked about, there's a lot in the name. Slow, of course, refers to what? Absolutely, they have slow myosin heads that produce a slow contraction. These are, so again, these muscle cells produce a slower weaker contraction than what we see in the fast glycolytic. However, these are oxidative. And again, as we talked about, there's a lot in a name. That means they make their ATP primarily using oxygen, right? Aerobically. That means they're very efficient. They may not be able to produce quick, powerful contractions, but they're very, very efficient. They can produce long contractions, right? They are going to be fatigue resistant. Right? Of course, to make their ATP aerobically, it's going to need lots of mitochondria. It's going to need a large blood supply. And it is going to need to store and hold on to a lot of oxygen. So it's going to have a large amount of myoglobin. Oops. And of course, if it has a large amount of myoglobin, these are the cells that we typically consider are quote unquote red muscle fibers. All right. So again, they're smaller and weaker, but they're much more efficient. They can be used for a prolonged period of time. They're fatigue resistant, right? Again, as we talked about before, how long can you stand for? Ever, right? And one of the reasons you can maintain your balance while you're standing up is that those postural muscles that help to stand you up have a large number of these slow oxidative fibers, right? They're not able to produce fast, powerful contractions jerking your torso through space, but we don't necessarily need to do that. We need to be able to hold ourselves in an upright condition. So holding ourselves up, right? Standing up. Holding and supporting your shoulder in place, right? Your joints, maintaining your joints, maintaining your posture. Things along those lines are what these are going to be used for. All right, and let's go ahead and clear my writing and then put the pretty pictures from your textbook in here. So like I said, right, you can stand forever. Everything is walking distance if you have the time, right? You can walk forever. You can stand forever. Because we have these slow oxidative fibers that may not be very powerful, may not be very fast, but they're very efficient, and so they are fatigue resistant. And so they can maintain a contraction for a much longer period of time. All right. That brings us to our third type. And again, we are still dealing with skeletal muscle cells. And those are our fast oxidative. Now, there's, of course, a lot in a name, but there's also a lot in the other information we've been talking about as well. Right? If the fast glycolytic were the biggest and the slow oxidative were the smallest, then guess where these fall? That's right, in between. These are going to be intermediate in size, and so that also means intermediate in strength. These are fast cells, so they have fast myosin heads, which of course means that they can produce a fast contraction. However, even though these are called fast oxidative, really, these cells are equally capable of producing ATP aerobically and anaerobically. Right. Equally able to do it both ways. So not surprisingly, they have an intermediate amount of mitochondria, medium blood supply, and an intermediate amount of myoglobin, right? Not enough to be red in color, but by the same token, more than enough to not be white. So not surprisingly, these cells are our pink muscle cells. Now, they are fast, but because they're able to successfully use oxygen to be able to produce ATP, these are also going to be fatigue resistant. This allows us to produce a moderate amount of activity with a moderate amount of strength and maintain it for a prolonged period of time. So this is things like slowly jogging or leisurely swimming in the pool or something along those lines. All right, so again, we erase what I've written, pretty pictures from the textbook. So like that jogging and that leisurely swimming. All right. Questions on these three types? All right, excellent. Now, there is one more issue. We've obviously, like we talked about in our turkey, we have the light meat and the dark meat. But does that mean we have light meat and dark meat as well? Well, sort of, but also not exactly, right? Our muscles, with one big exception, are all mixed muscles. So let's go here. Actually, I'll do it here. I can fit it in here. Make it a little messier. So here's the magic word. Almost all muscles in our body are mixed muscles, meaning that they're a combination of all three types. Now, does that mean that they're all equal amounts? Right? No, not equal amounts. But almost all the muscles in the body are mixed muscles. There's really only one exception. The only exception are the muscles that move your eyes. Right? If right now, I know you're staring at the screen right now, but look up, look down, look left, look right. That ability to move your eye within the orbit of your skull is done by 6 muscles that are located in there. You're not actually, you didn't have to learn them for this exam, but I will tell you, you are going to actually learn them in the nervous system. So we will get to learn 6 more new muscles when we get to the nervous system. But for now, you don't have to know them. But know that there are 6 muscles inside each of the orbits of your eye to move your eyeball around. Those muscles in your eye are the only pure muscles in the body, and they are pure, fast glycolytic. But every other muscle in your body is a mix of both. All right? And that's what we see nicely. We go ahead and erase this in this pretty picture from this one. I don't think is from your textbook. Actually, this one I think is from your textbook. I have another one that I like as well. What we have here is a cross section through a fascicle. And so what we're seeing here are individual skeletal muscle cells that are cut in a cross section. And what's cool is what they've done here is that they have used a stain that specifically stains for myoglobin. So notice these two have a massive amount of myoglobin, and these are our red cells, our slow oxidative. Notice this cell has almost no myoglobin in it. It's very, very light in color. And so this is our fast glycolytic. And notice the majority of the cells in this fascicle have an intermediate amount of myoglobin. These are those fast oxidative. Here, this one's not from your textbook, but I love this picture. I think it's really pretty. I want this to be a wallpaper in my house. But what we have here is here we can see many fascicles within a muscle. And again, we see the same thing. We see some that are very light in color, some that are more intermediate in color, some that are very dark. Here we see 3 of them right together. So again, with fast glycolytic, slow oxidative, and fast oxidative. We have these 3 types. And we differ in where they're located. We mentioned Thanksgiving is right around the corner, right? But how many people, and again, you can use under participation, there's a yes or a no. How many people here have eaten duck before? Give me a yes or a no as to whether or not you have eaten duck. In particular, duck breast. Come on, I want more than 5 answers. Let's go. Yes or no? Have you eaten duck? Don't make me start calling people out by name. Eric, Ashley, Edward. No, I want it on the participation. I want you to click yes or no on there so I can see it more easily. I don't want to have to read it. Make my life easy. It's okay if it's a long time ago. Even if it was a long time ago, that counts. There you go. That's a much better mix. Excellent, excellent, excellent. All right, good. About half, it looks like, of the people that have answered. That's great. So, especially when you eat the duck breast, and now you can type it out if you don't want to speak. Does duck breast look more like chicken breast, or does it look more like chicken thigh? For those of you who've had it, it looks more like thigh. It's actually dark meat, exactly. And again, think about how these animals get around. That chicken, that turkey, right? That turkey on Thanksgiving primarily walks. So he has to stand all day long and walk all day long. So having those slow oxidative fibers that allow him to stand for long periods of time are going to be in his legs and in his thighs, right? A turkey is capable of flight, right? But they don't, it's not their primary way of getting around. So they can produce a fast, powerful contraction with their wings to get over a fence or to hop on top of a roof or something along those lines, but it's not their primary way of getting around. So they have more fast glycolytic muscles in their chest, in their wings, to be able to produce those powerful contractions, but it's not the way they get around. Ducks are the opposite. Ducks have dark meat in their breast because their primary way of getting around is flight. So they want to be able to sustain those long contractions, smooth contractions, right? They're not flapping their wings like a hummingbird to get from place to place, but they need to be able to maintain contractions of their wings to be able to get to where they're going. So in that case, it is their chest that is got the slow oxidative fibers in them. So we're different. And if you think about it, when it comes to a duck or a turkey, which one are we? We're turkeys, right? Our primary way is to walk on our legs. As we talked about, how long can you stand for? When you stand, you're holding all of your weight upright in your legs and in your thighs, but they have those slow oxidative fibers that are able to do it, right? I think we asked this question before, but I'll ask it again. How long can you plank for? Hold yourself in that planking or that push-up position for? 3 minutes, 10 seconds. There you go. Every 3 minutes. Perfect. 10 seconds. I like it. Yeah, absolutely. Some far, far less. And if you think about it, when you're planking, you're not even holding all of your weight up. So again, our chest, our arms, we can throw that powerful punch. We can throw that ball really far. We can make those fast, powerful, intense contractions, but they can't be sustained. So our legs and thigh have much more of the slow oxidative and much more of the fast glycolytic in our arms and in our chest. All right. Now that doesn't mean that. Well, let's go back. So next question: if we all worked out the exact same every day, so all of us from now to the end of the semester, we all grabbed 15-pound weights. We all did 10 curls with it 3 times a day, every day from now to the end of the semester. All of our arms would get bigger as a result of that. But would all of our arms get the exact same size bigger? Would all of our muscles get the same size as a result of that? No. And the primary reason for that is genetics. We all have these three different types of skeletal muscle fibers in our muscles, in our arms, but some of us have different amounts. And that different amounts is primarily determined genetically. So those of us that have more of the fast glycolytic, when we're doing those bicep curls, are going to gain more benefit from them. Whereas those that have fewer are not going to see as much increase in size. All right. And again, arms are a great way to think of it, but I think one of my favorite ways to think of this is in terms of runner. Right. I know the Olympics are going to be put off for another year, right? So we're going to have to wait another year before we can see Usain Bolt, right, who's a massive sprinter, and we can see Meb, who's a marathon runner, right? These are two world-class professional athletes, but when you look at them, they look very, very different. And the reason for that is that they have been specializing different muscles within their body. Usain Bolt is all about the fast glycolytic, right? Fast, intense, powerful contractions that only last for a brief period of time. Where Meb, is that marathon runner, is slow and steady, but for mile after mile after mile after mile. Like I said, both are world-class athletes at the top of their games. But Usain Bolt can't just decide tomorrow that he wants to become a marathon runner, right? Well, he could decide tomorrow, but tomorrow he's not going to be very good. And even after years of training, he may not be very good. Now, the one thing that we can do, so the other last question is, what muscles you have is determined genetically, but there is a limited ability to be able to modify those. In particular, it is easier to modify our fast glycolytic into our fast oxidative. So it is possible with training to be able to modify some of what we have. But again, a lot of what we have is determined genetically. All right. Questions on that? All right, excellent. With that, then, we are now truly and thankfully done with everything having to do with the skeletal system, skeletal muscles. So we are done with our skeletal muscles. It is a teeny bit early, but this is a good natural stopping point. So let's go ahead and stop here, take our 10-minute break. So we'll come back at 1 o'clock. And at 1 o'clock, so let's actually go back so we're at least on a pre-picture. Hold on. Go there. Excellent. So we will return at 1, and at 1, I will restart the recording. Excellent. So let's put that in the corner to remind us. Take a quick 10-minute break, and then when we come back, we will then briefly, again, we're not going to obviously spend nearly as much time on smooth muscle and cardiac muscle, but we do need to talk about them to see how they're similar and how they are different. All right. So any questions before we take our break? All right. Then I will see you back here at 1 o'clock. All righty. Let's go ahead and get started. I see there's a great question. Let me answer that real quickly before we dive back into the new material. With the muscle histology, again, you don't have to turn in the pictures that you're drawing. That is just your study guide to help you. What I would say is that the histology books actually do a pretty good job, at least the one we have, the LaBeouf does a pretty good job of labeling things. What you might want to do is, it may not necessarily label everything on every particular slide, but especially if you start at the beginning of a section and also read the descriptions, they do a really nice job of doing things that way. While it is intermittent in how good it is, if you also look at the Cosumnes River College Visual Anatomy, their histology on their list is pretty decent, and they've got the slides where it shows both the slide and then also the slide with tags. So that might be a good place to go. Let's actually take a peek at that again. Give me a second. So, there we go. So, oops, didn't click it. Again, from your module, there is the link that gets us to the Cosumnes River College's Visual Anatomy Lab. Here, if we come to histology, while it doesn't have a massive collection, it does do a nice job of showing some pictures and then also labeling them as well. So you can see a picture and then see the label. So that could be something that could be worthwhile. So you could use this to be able to distinguish some of the characteristics of that. So that could be something that could help you with your histology, as well as your textbook. Also, remember your personal anatomy lab has, no, I keep saying personal, the professional anatomy lab also has a histology component, and that does a nice job of describing and labeling things as well. So in your modified master in A&P, there's that professional anatomy lab in the study areas and the lab tools. That would have an excellent source for histology things as well. All right. Great question. Any others? Alrighty, got everything up and going. So then let's go on and pick up where we left off. Annotation tools. Sorry, it takes me a minute to get, when I bounce between different screens, it takes me a minute to get everything up and running again. Oops, didn't want to do that. All right, oops. So let's talk about our smooth muscle tissue then. With our smooth muscle tissue, we have that microscopic anatomy that we are familiar with and that we know. Again, remember when we identify, and that's the one thing, remember also when you look at that histology list, not only are you responsible for identifying all of these characteristics for that particular tissue, but once you identify a tissue like smooth muscle, you also need to be able to identify the characteristics. So I could also ask you for the characteristics of that tissue as well. And as we talked about, smooth muscle is small, it is spindle shaped. Again, spindle shape, remember, means that it has kind of a centrally located belly and an elongated, tapered tails on both sides. It has a centrally located single nucleus found at the center within the cell, large cell body of that smooth muscle cell. Just a single nucleus, different from our skeletal muscle. And obviously, as the name indicates, one of the big differences in it is that it is unstriated. There are no striations to it. All right. Being a single cell, remember, as we saw, our skeletal muscle cells are composite cells where hundreds of cells have come together to form it. And that made repair challenging. Instead, we have to rely on those satellite cells to help to replace or repair damaged muscle cells. Whereas smooth muscle, being a single, simple cell, it is much easier for it to divide to produce new cells to replace those that have been damaged. So as we talked about, it has the highest rate of repair. We also talked about how most smooth muscle, as we see here, is within the walls of a hollow organ. So that was one of the characteristics that we learned about it as well. And again, none of this is new information. It may just feel like that because we talked about it a long, long time ago. Also, as we see nicely from this illustration, typically smooth muscle is arranged in layers. Notice, and I'll go back to the highlighter. There is the innermost layer is a circular layer. That circular layer, when it contracts, is going to change the diameter of this organ so we can increase or decrease the size of the lumen. Whereas a longitudinal layer runs the length of the organ. So when it contracts, it makes the organ shorter. In fact, a smooth muscle arranged like this does a very special and important thing. We can have alternating contractions of these layers where the longitudinal shortens the organ, and then the circular squeezes the lumen, and then it shortens and squeezes and shortens and squeezes. And with that rhythmic alternating contraction of the smooth muscle layers, we are able to propel substances through our body. So that cheeseburger you had for breakfast works its way through the digestive system. Your urine works through your urinary tract. There are a lot of organs that use these rhythmic contractions, what we call peristalsis. And that peristalsis is one of the ways that we are able to move structures through our hollow organs using those smooth muscle contractions. When we talk about the microscopic anatomy, as we've talked about, it is non-striated and it is involuntarily, we do not consciously control it. What that means, remember, as we talked about, let me get that out of my way, is that this means that it is controlled by our autonomic nervous system and by hormones. Unlike our skeletal muscle, which of course we know is controlled by our somatic motor nervous system. And I will save you the drama. Remember how we talked about how there are 3 specific types of skeletal muscle: fast glycolytic, fast oxidative, and slow oxidative. We have 2 specific types of smooth muscle. They are what we call single unit or visceral smooth muscle. This is called a single unit because it works together. As a single unit. That's really big. The cells work together as a single unit. And so that's why it's called single unit. And it's also called visceral because primarily this is the one that are found within our hollow organs, like our stomach. What allows them to work together as a single unit are those gap junctions. Remember, we talked about how gap junctions are fluid-filled holes between the cells that allow the cells to communicate with each other. So if there's an influx in sodium in one cell, that can spread to the other, and so an action potential can spread quickly from one cell to another, so it all works together. And one of the really interesting things about the smooth unit is there is some autorhythmicity. Autorhythmicity means that it is capable of producing its own action potentials. Again, your stomach is a great example of this. We'll talk about this in great detail when you get to the digestive system in 431. But for instance, when you have that cheeseburger for breakfast, what happens is the cheeseburger gets into your mouth, swallowed down your gullet and into your stomach. And once it gets into the stomach, it stretches your stomach. Your stomach has stretch receptors that perceive that stretch. And then your stomach actually basically has its own neural network called a plexus that basically takes that information, processes that information, and says, hey, something is stretching out the stomach. We need to do something about it. So let's get the stomach to contract. And it causes the smooth muscle of the stomach to contract to churn that food inside of it. And all of that decision was made outside of your own nervous system, made outside of your conscious, but even your autonomic nervous system. The stomach itself makes that decision. And so it has some of that functionality to it. However, remember, we also learned that not all smooth muscle is in the hollow organs, like our erector pili muscles. That type of smooth muscle is typically what we call multi-unit. These have to be communicated to independently, just like we see with our skeletal muscle cells. So each one is individually innervated, and they're forming into fascicles, and they form bundles like our erector pili muscle used to make your hair stand up on end. And here we have the pretty picture. Again, here these all have gap junctions for our single unit. So it takes only a couple inputs from our autonomic nervous system to get this whole thing to work together. Whereas here is a very irregular bundle of our multi-unit smooth muscle, like that erector pili muscle. All right. However, where our smooth muscle is probably most different from our skeletal muscle is in its microscopic anatomy. Again, because it is smooth muscle, and we'll emphasize this up here in red, in big, right? No stripes. And really what that means is no sarcomeres. Right? There are no myofibrils, there are no sarcomeres. Right? Sarcomere is those Z discs and the myosin and the actin and all those things that are perfectly arranged and aligned so that we understand how a skeletal muscle contracts. And if there's no stripes, oops, I hope I spell stripes right, and no sarcomeres, then obviously it uses a very different mechanism for contraction. And so to see that, we have to look at the anatomy a little bit more. Now I like that diamond shape. I'm going to totally steal that. So I can draw my own skeletal muscle, smooth muscle spindle cell here, like the one we see down below. And we can talk about some of the anatomical distinctions of it. One of the things that I want to point out, I'm not going to be able to necessarily draw all of these things, but I can cheat a little bit. Where's my stamps? I will do it with stars. So give me a color, pink. That's not pink, but oh well. Notice on the surface of it, we have these structures that are known as the caveolae. Caveolae are basically invaginations within the plasma membrane, kind of like a transverse tubule, but it doesn't go all the way down. Caveolae means little cave, and that's literally what this is. Little indentations in, oh, and that's way too big. Let's make that black. Let's make it small. Too small. Little indentations in the plasma membrane with a massive number of calcium channels. Our smooth muscle has basically little to no sarcoplasmic reticulum. That means it relies much more heavily on extracellular calcium. And these caveolae are how they get it. So these caveolae allow our, allow our calcium to come in from outside. So to move those regulatory proteins, we need those caveolae to allow all this massive amount of calcium to come in. So we said no sarcomers, no myofibrils, but we still are going to need contractile proteins. Right? And of course, those two contractile proteins are myosin and actin. So it does have myosin and actin, but in a different arrangement. Right? There are no Z discs, right, to be able to anchor our actin, but we still need to anchor our actin. So as you can see here inside of it, and I'll just draw a couple of these, one there, and one there, and one there, and one there, and then I need to draw a couple more here, and here, and here. There we go. We still need some anchors for our actin. And these anchors for our actin instead, so no Z disc, instead, it has what we call dense bodies. And these dense bodies connect to the plasma membrane. So here we have these dense bodies, and those are the pink things. So right here, we'll just write DB to remind us of that. In between our dense bodies are structures. Oh, I'm going to use the highlighter for this. In between our dense bodies are extensions of the cytoskeleton that are our intermediate filaments. So our dense bodies are connected by intermediate filaments. And then in between these is where we have our myosin and our actin. So let's go ahead and use blue for our myosin. Here is our myosin, basically in between the dense bodies. Our myosin, like the myosin inside of our skeletal muscle, has heads on it. But one of the big ways it's different is the myosin heads go the entire length of the muscle cell. And what's interesting, the filament. And what's interesting is that unlike before, where the ones on the right face right and the ones on the left face left, on one side of the myosin, all the heads point in one direction. And on the opposite side, all of them point in the opposite direction. And I know I'm making these myosin heads way too long, but it's all right. Hopefully you get the idea. Now, of course, these are going to need to connect to our actin, and it is the actin that is anchored by our dense body. So here's an actin on our dense body here. Here's an actin on our dense body here. And so if you think about what happens when our muscle contracts, when the muscle contracts, all myosin is going to perform their power strokes, and it pulls this actin this way, which of course pulls this dense body that way as well. Whereas this one down here pulls the actin this way, which pulls this dense body that way. So notice what happens is our two dense bodies are pulled towards each other. But notice our dense bodies are connected to other dense bodies by these intermediate filaments. So when this one gets pulled, this dense body gets pulled towards that dense body, and these get pulled towards this one. And when this one's being pulled, this is going to get pulled towards that, and so on and so forth. And notice these are not linear movements. When this muscle cell contracts, what happens is it shortens, it is getting shorter, but notice also the muscle cell twists. It is going to twist and turn when it contracts. Now, again, I've done a very superficial job of explaining this, but even with just a simple explanation, you can see that this is not necessarily a very efficient way to do this. It is going to produce slow and very weak contractions, because this is not an efficient way to do this. However, because of the twisting and turning, what happens is we produce slow, weak contractions, but it can be sustained for a much, much longer period of time. Only a little bit of energy is needed to maintain these contractions. So while it produces slow contractions, these slow contractions can last for a very long time. They are very fatigue resistant. Oops. Very, very fatigue resistant. So again, if you think about, right, when your stomach is contracting around that cheeseburger you had for breakfast, we don't necessarily need to be producing big, huge, massive, powerful contractions to pulverize that cheeseburger inside of you. We want slow, sustained contractions to just churn it up, right, and move these things. So it allows for very prolonged, very fatigue resistant, slow, weak, but sustained contractions, making it a more efficient pump-like action. All right, let me go ahead and take a pretty picture of this so I can share that on the site. And then we can look at the pretty pictures from your textbook to continue that exploration. So let's go ahead and clear that now. Like I said, it has our myosin and our actin. It has those thin and thick filaments, but no sarcomeres. Right, also no transverse tubules. Instead, it has the caveolae and very little sarcoplasmic reticulum. So it does not rely on calcium from inside the cell. It relies on extracellular calcium, calcium from outside the cell. No Z discs, but we still need an anchor point for our actin. And again, it doesn't really, it doesn't anchor the sarcolemma. Oh, it attaches to the sarcolemma, to the plasma membrane, absolutely. Attaches the plasma membrane to the sarcolemma and is also the attachment point for our actin. And then we have those intermediate filaments that connect the dense bodies together. Our actin and myosin line up between those dense bodies. And here you can see it produces that slow, inefficient contraction where we get this shortening, but also twisting and bulging of this muscle. Like I said, it is very inefficient. This contraction can occur 30 times slower than a contraction for our skeletal muscle. But because of all this twisting, because of all these interconnections from the intermediate filaments, only a small amount of ATP is required to maintain it. So these contractions can be maintained for a much, much longer period of time. And again, as we talked about, this is used for the process of peristalsis, those rhythmic contractions to move it through space. All right. Questions on our smooth muscle? All right, excellent. From our smooth muscle then, we need to, oops, what's going on? There we go. Talk about our cardiac muscle. Again, when we think of the characteristics that we know about it, it is branched. It is autorhythmic, capable of producing its own action potentials. It has these, and here's the, right, these special cell junctions between them, those intercalated discs that holds this branching cells together. Inside those intercalated discs, remember, as we talk about, there are a large number of gap junctions and also the desmosomes to hold them together. Notice they do a nice job here on the illustration, oops, of giving us the gap junctions where it holds it together, but it forgets to show us those desmosomes. Those desmosomes are going to be important for holding that together as well. Cardiac muscle has a single nucleus, but it is striated. So that means it does have myofibrils. It does have sarcomeres. And that means that our muscle contraction process is very similar. In fact, the how, whoops, it contracts is identical. Right, that contractile cycle, the moving the troponin, the moving the tropomyosin, all of that is identical, but the actual action potential that is produced, and therefore the contraction, the twitch that is produced, are going to be different. So that's where they're going to be different. They are different in the action potential, or let's be more specific, the muscle action potential, and the twitch that is produced. All right, and we'll talk about those in just a second. Remember, smooth muscle has the highest rate of regeneration. Skeletal muscle has a little bit of regeneration, but remember, our cardiac muscle has no regeneration to it. And of course, cardiac muscle tissue is found only in the heart. When we look at the microscopic anatomy, as I mentioned, there are some similarities to what we saw in our skeletal muscle. We have myofibrils. Oops. We have transverse tubules. We have a sarcoplasmic reticulum. Those are things that are definitely similar, right? However, notice there's no terminal cisternae. There's no triads. While our cardiac muscle cell does rely on intracellular calcium, it also has a fair amount of extracellular calcium that it has to it as well. So it has a much less extensive sarcoplasmic reticulum. It has no triads, and it does rely on some extracellular calcium. And again, while the contraction itself is identical, the action potential and the twitch are going to be different. All right, let's talk about that. Let's do this. I'm going to go ahead. Let's go ahead and clear that. Go back to our whiteboard. Need a line. Perfect. All right. Let's talk about the things we know first. One of the things we know is a skeletal muscle action potential. It's something we've talked about and something we know. It has more calcium gated channels. So yes, and we'll talk about that. Great question. It does have more of the, no, not more chemically gated. It has more voltage gated calcium channels. All right, let's go ahead and draw a graph of what we think when we think in terms of our skeletal muscle action potential. Of course, this is something that is time. This is something that is voltage. Right? We know that this cell has a resting membrane potential where it'll happily stay at forever. And what do we say the resting membrane potential was typically? Anyone remember? -70, excellent. -70 millivolts, perfect, right? That resting membrane potential where it'll stay it forever. However, if we stimulate or when we stimulate it, what happens is we get a rapid depolarization followed by a rapid repolarization back to rest, right? So let's write that out. It is a rapid depolarization, right, which means it's getting more positive, followed immediately by a rapid repolarization, which again, remember, means going back to rest. Followed by a rapid repolarization. And this whole thing typically takes, I don't know, 1 to 2 milliseconds. It is super, super fast. All right? We know that. Of course, we also know that that skeletal muscle action potential produces a skeletal muscle twitch. Again, we need to draw that twitch. We can draw our, let's cheat, hold on. I want you here, out of my way. And of course here, still time on the bottom. It is tension over here on the side. And we know that our twitch was slightly offset, right? We have the production, and I'm just going to remind us here, this is where we produced our muscle action potential. And when that occurs, remember our twitch has 3 stages to it. And I'm going to need to change the color of it so we can see it. The first phase is that latent period. That is followed by the contractile period, which is followed by the relaxation period. And again, we can distinguish these 3. Our latent period or latent phase is the part where the tension does not change, the tension stays 0. Our contractile phase is when tension is increasing. And then our relaxation phase is when tension goes down. So again, latent phase, tension equals 0. Here, I need a T there. And I need a T there. To remind us that during the contractile phase, our tension is going up. And during our relaxing phase, the tension is going down. And then, of course, once it reaches 0 again, then we are back at rest. Right? There's a little bit more. Obviously, if I ask you to describe a twitch, you need to give me a little bit more detail than that. But that's a good reminder of what it is, that it has those 3 phases. You obviously need to tell me what's going on, why the tension is changing in these 3. But hopefully some of that, because you've been studying this, is vaguely familiar to you. If not, well, the good news is you have 2 days before the exam. Excellent. So right there on the board, we have absolutely no new information. This is stuff that we already know and we already understand. So what we need to compare it to is what's going on in a cardiac muscle cell. Oh, one last thing. Timing. Remember, this twitch in our skeletal muscle cell lasts, as we talked about, from, I don't know, let's say around, like that, 70 to 100 milliseconds. All right. That seems a good range. There's some that can be faster, especially some of the fast glycolytic can be faster, but that's a good range. Heck, we can even say, let's say 20 to 100 milliseconds. Excellent. All right, let's compare this then to what we are going to learn about our cardiac muscle action potential. Our cardiac muscle action potential is much longer than a skeletal muscle action potential. And we'll see why when we draw it here. Let's go ahead and put, there we go. And again, it's going to be the same thing. Over here we have voltage. Down here we have time. Can you guys see where I wrote time? It's hard for me to tell what you guys can see and you can't. Okay, perfect, excellent. All right. What happens is that it starts similar to what we saw in our skeletal muscle action potential. And I'm going to offset it a little bit just so I have more time, space to write. What happens, the first thing that occurs is we get a rapid depolarization. Just like we saw with our skeletal muscle. And just like it, this is actually due to sodium influx. It is sodium that comes into the cell, makes the cell more positive. And that's exactly what happens here in our skeletal muscle. So we have this huge influx of sodium, and because of that, we get a rapid depolarization. So far, everything is the same. All right. However, what happens now is that rather than our skeletal muscle, which immediately depolarizes afterwards, what happens here with our cardiac muscle is we have a prolonged depolarization. This prolonged depolarization is in primarily, due to the influx of calcium. Calcium, remember, comes in from outside of the cell. Our cardiac muscle relies on calcium from outside the cell, whereas all the calcium that is used for a skeletal muscle cell is on the inside. That influx of calcium leaves the cell depolarized for a prolonged period of time, producing what is called the plateau stage. Eventually, though, the cell is going to repolarize back to rest, and it's going to repolarize primarily by the efflux of potassium. So potassium leaves the cell, and that's in my way. And that potassium leaving the cell brings the cell back to rest. That is also what happens up here in our skeletal muscle. Over here, it is sodium that causes the depolarization, and it is potassium that causes the repolarization. That part is the same. But what's different about our cardiac muscle is this prolonged depolarization in the middle. And in fact, the muscle action potential in our cardiac muscle can be as much as 15 times longer than in skeletal. So it produces a massively, massively long action potential. All right, questions on that? Now, the good news is, just like in skeletal muscle, one muscle action potential produces one twitch, and the same thing is true in our cardiac muscle. One cardiac muscle action potential produces a cardiac muscle twitch. Another piece of good news is it has the exact same 3 phases. Again, it still has those regulatory proteins that need to be moved. Oops, don't know what just happened there. Good enough. So again, it is going to be time down here, tension over here. We are again going to get blue. Our muscle action potential is produced. And from that muscle action potential, we are going to get those same 3 phases. We're going to get a latent phase, we are going to get a contractile phase, and we are going to get a relaxation phase. Now, obviously, I've made that a little more steep than it necessarily needs to be, but I ran out of room, and we all know my drawing and writing skills, so it's fine. We get the idea. The point is, it produces the same twitch that is going to have those same 3 phases. We are still going to have a latent period, a contractile phase, latent phase, contractile phase, relaxation phase, right? Here, the tension is still equal to 0. Here, tension is increasing. Here, tension is decreasing. Again, all of that is the same. What is different is the timing. The big difference is the timing of this. Here, the twitch can last over 200 milliseconds. It produces a much longer contraction. The rest, yes, for these cells, our resting membrane potential is similar. Yes, I realized my drawing, I didn't quite show that. But yes, for argument's sake, great question. And again, this is more of a simple sky is blue type of answer. Trust me, when we get to the cardiovascular system in 431, we'll learn this in much, much more detail. You guys are getting the incredibly sky is blue version of this. But yes, these contractile cardiac muscle cells we're talking about have the same resting membrane potential for us. So much longer action potential produces a much longer twitch, which again, if you think about it, makes sense for the function. The function of the heart is to act as a pump, right? I'm sure many of you have used a pump before to blow up like an air mattress, right? And again, I don't mean the kind that you plug in and push a button, right? I mean actually using that hand pump up and down and up and down. And if you're going to use that hand pump, right, do you want to make a whole bunch of really fast, rapid contractions with your hands to do that? Or do you want long, full, sustained pumps of it? And again, you want those long, full, sustained pumps, absolutely. It makes it a more efficient pumping action. And that's what we have here. This difference in the length produces a longer sustained contraction to efficiently pump the blood out. Now, there is one more big difference between our twitches here and our action potentials here in our cardiac and skeletal muscle cell. If you remember, as we talked about with our action potentials, let's not talk about twitch, let's talk about action potentials. If you remember, we said that when you produced an action potential, right, so we produce that action potential here. If you remember, we said there is a, oops, let's not do that, undo that. There is a brief period of time where it cannot produce an action potential again. It loses its irritability. Does anybody remember what we called that period of time? There's a brief period of time when our muscle cell got excited where it no longer could be excited. We couldn't stimulate it a second time. There you go, excellent. It was called the refractory period. And if you remember, our refractory period for our skeletal muscle cell was somewhere between 1 and 5 milliseconds. So if you think about it, we produce that action potential. We have to wait 5 milliseconds to produce a second action potential. But if you think about where we are in the twitch at that period of time, in our twitch, 5 milliseconds is only taking us to about right here, right? We're barely starting to produce tension at that point. And that's why, as we said, if we produce a second action potential at this time, we produce a second twitch, we add those two twitches together. And what do we call that process is where we add the twitches together to get this big, long, sustained, locked up contraction? What did we call that again? The process? No, it wasn't Treppy, it was the other one. Wave summation, absolutely, where we're adding the waves together. We were able to wave summate. So because, notice, this has a short refractory period, we can wave summate, and that allows us to, remember, tetanize the muscle, either with a fused or an unfused tetanus, right? We can produce that long, sustained, locked up contraction. Now here with our cardiac muscle cell, that refractory period is over 250 milliseconds. Right. Not only is that well past where this action potential is, but if you think about it from where we produce this, remember our twitch only lasts for 200, so it's well past that. So notice with such a long refractory period, we cannot wave summate, right? Which also means we cannot tetanize the muscle, which if you think about it, is a good thing. Do we really want to be able to tetanize our cardiac muscle, have our cardiac muscle lock up into a sustained contraction that we can't control? No, absolutely not. So the fact that it has a much longer refractory period is actually an absolutely good thing. We get those slow, sustained, long contractions of it where it's not going to lock up, it's not going to tetanize, and that way we can maintain that smooth rhythm of that beating heart. All righty. Let me go ahead and take a picture of our whiteboard for that so that we can look at the pretty pictures for that. And then we'll go through all of the stuff that we talked about again with all the pretty pictures from our textbook and from everything else. So I need that back. And then I need that. And I need that. There we go. All right, excellent. So any questions on this? So again, remember, complete tetanus is, oh, I see what you're saying. So the difference between a complete tetanus of a skeletal muscle and this prolonged contraction of the cardiac muscle is probably the intensity of it. The amount of tension that is being produced in a tetanized skeletal muscle is probably greater than a single cardiac muscle. However, you've got the right idea in that the timing of them are similar. They both last for about the same amount of time, although a tetanized contraction of your muscle could last longer if we needed it to, right? This we don't want. Here we want to maintain a smooth rhythm. How long you tetanize that muscle depends, right? If you are holding up, a file cabinet that is about to fall on the baby, you need to maintain that complete tetanus in that muscle for as long as you possibly can until someone can get that baby out of there. Right? We don't want our heart muscle locking up in a contraction that lasts as long as it possibly can, right? Because something is artificially stimulating it. That would be something that would be really, really bad, and we wouldn't want that to happen. Right? And so we can avoid that. That's interesting. Okay. So yeah, but as far as the intensity of it, the strength of them, they're probably similar. Great question. Any others? All right, then let's switch back to our lecture screen. There we go. And go through this stuff again. So like I said, the big difference is between skeletal muscle and cardiac muscle is that our muscle action potential and our cardiac muscle is much, much longer. As we said, it can be up to 15 times longer. And that's because it has that plateau stage because of that prolonged calcium that is being delivered. The contraction lengths are much, much different, 20 to 100 milliseconds in skeletal muscle, whereas our cardiac muscle, a single twitch, lasts over 200 milliseconds. But one of the big differences is in their refractory period. Skeletal muscle has a very short refractory period, which is what allows it to be tetanized, is what allows us to wave summate, whereas the refractory period for our cardiac muscle is longer than the contraction. So we only get one contraction at a time. We can't add waves together to dramatically increase the tension because we don't want to do that. We don't want to increase the tension dramatically and uncontrollably by tetanizing the muscle. We want smooth, sustained, powerful, continuous contractions. And then, of course, as we talked about, our cardiac muscle has gap junctions and has special cells that are autorhythmic, so it produces its own action potential and spreads it through it. So it does not need motor units. It doesn't need the motor units. Instead, all it needs is that one special cell that's going to make and produce an action potential and spread it so that it works together. Whereas our skeletal muscle needs those somatic motor neurons to stimulate it. And there you go. Like I said, they're those special 1% of our cardiac muscle cells that are autorhythmic. capable of producing their own action potential. If we were on campus, I will tell you quite frankly, you would have learned the contractile process or the excitation process of these autorhythmic cells. I think it's actually in your lecture slides, but because of the awkwardness of this and it's a challenging concept, and more importantly, simply because you will do it again when we get to the cardiovascular system and get to the heart in 431. So you will, I know for certain 100% get that information. So I'm not going to make you do it now. All right. What that means is that is it. Again, your book's got some nice tables summing up the similarities and the differences, the uniqueness characteristics of our skeletal, cardiac, and smooth muscle. Make sure you've looked at that. Make sure you understand that. Make sure you're looking at the histology of these and understanding all of that information. And with that, that is everything we need to do from a lecture standpoint for today. All right. And that is definitely the good news of that. The bad news is we still have a lot of information to talk about because we haven't talked much at all about the legs. So we need to talk a little bit more about that and some of the important things that we need to do for our origins, insertions, and actions. So we still have some lab stuff we need to talk about. But let's go ahead. This is a good natural breaking point. So any questions on cardiac muscle versus skeletal muscle before we take our next break? All right, dead silence means that you've all mastered this material and I can make the test tougher. Awesome, I love that. Excellent, so let's go ahead and take our next break. Looks like it's 5 minutes till 2. Let's return at 2:05 then. And again, I will restart the recording at that time. And like I said, we will switch gears at that point. I'm teasing. The test is hard enough. Actually, I don't, like I said, I don't know how this test will relate hardness-wise, in all honesty, compared to the previous ones. I know there's a lot more information on my end that went into it because, like I said, for instance, when I write this and where I'm labeling the models, I don't have to label every single model 4 times to ask those 4 questions because not every single question is going to be asked for all 4 questions. But the same thing's going to happen on this. Like I said, I literally made over 300 lab exam questions for this exam. But those weren't all for you. You're not going to have to take all 300. You're just going to have to take 80, the same way you would have had to take 80 in the exam on campus. On campus, when we do this, it's usually somewhere between, I don't know, 80 and 85. So it's a similar number of questions. So how much information you're responsible for is pretty much the same. The big difference is the randomness of it. Right? All of you are getting bone pictures. All of you are getting bodybuilder pictures. All of you are getting muscle pictures with origins and insertions and actions, but I don't know how many. One person may get more actions. One person may get more origins. One person may get more insertions. And I don't necessarily know if one of those is easier or harder than the others. There's just a lot more fluidity to it. All right. Hello, would you be able to go over what we need to, yeah, sure, we can talk about those types of things, the modification of the neuromuscular junction again. We can do that, but let's wait for that at the end. So at the end, we should be able to finish the lab stuff, because again, I'm not going to go over all of it for you. Again, one of the onuses with going online is that a lot more of the onus for mastering the lab material is on you and your responsibility as students, so we will talk about that together. But I do anticipate having time to answer any questions, like a review type thing to help you with any information that can help you to be successful on the exam itself. And so asking questions about modification of the neuromuscular junction is a perfect example of the kind of question to ask when we get to that. So yes, I do anticipate having time to do that today as well. All right, any other questions? All right, excellent. Let's go ahead. Like I said, take a 10-minute break. My watch says 1:57. So let's make that 2:07 so everybody gets the full 10 minutes, and then we'll come back and we will pick up from there. So let's pause the recording and get started. So the last little bit of information we need to deal with, and that's the last little bit you need for the exam, is to do some discussion on some of the leg muscles. I don't think we got to any of the leg muscles in the last class. Can you guys remind me, did we talk about any of the leg muscles? Anyone remember? It's been a while. Yeah, I didn't think we had. Okay, excellent. Again, like last time, I'm not going to have the opportunity to go through all of them on the list, but there are some that I do want to point out to you to make sure to emphasize and talk about some of the differences or some of the key things that I find is sometimes challenging or confusing to individuals. So I think that that can be worthwhile. Starting first with here, with these two muscles, the gluteus maximus and the gluteus medius. If we take a bit of a closer look, gluteus maximus is obviously what most people think of when they think of, right, your posterior. And again, not surprisingly, because it is a posterior muscle. There are a couple things about it that I want to make sure to emphasize. Notice that its origin is very much on the posterior. As we can see, it's on the posterior part of the iliac crest and down the sacrum and down the coccyx. So it has a very posterior and also medial origin to it. And let me cheat and go ahead and bring up my list. And notice its insertion is also posterior. Notice our insertion is that gluteal tuberosity, that kind of V-shaped roughened patch on the posterior part of the femur. And again, if we cheat, we can actually look at our bone picture. It's not a cheat, but we can look at our bone picture as we've been doing before, where we were talking about drawing these things. Again, as we saw, the origin, as I mentioned, is along the dorsal part of the crest and along the sacrum and the coccyx. And again, because I'm using the highlighter, it's probably a little bigger than it needs to be, but we can see that okay. And its insertion, as I see, the bony insertion on this, as you can see, is onto the gluteal tuberosity, which is that, oops, wrong thing. You're supposed to be blue. And actually, let's use the line so we can be more precise. It's kind of that roughened V-shape region on the superior posterior surface of the diaphysis of the femur. So we have that there. However, if we look at the illustration again, what we can notice is that we can absolutely see the fascicles that are coming down and attaching to the femur. But notice a lot of them attach into this white stripe that is on the lateral aspect of the leg. And let's go back a picture. Notice this white stripe goes all the way from the iliac crest all the way down to the lateral aspect, to the lateral aspect of the tibia. And so not surprisingly, this white connective tissue structure, one of the structures on your list, is the iliotibial band. This iliotibial band, remember we talked about, we saw it when we actually did our cadaver. I'm sorry, do I need to close the door? Forgot to close my office door when I went back in there, getting dirty looks from my kids. All right, the iliotibial band, remember, is an extension of that fascia lata. Remember that fascia lata is, now I will use the highlighter. That fascia lata is a big, huge connective tissue sheath that wraps around the entire leg. We actually saw that when we had the cadaver in class. We saw there was this fibrous connective tissue wrap that wrapped around it, and it acts basically as a pressure cuff to help to put tension, holding and stabilizing the muscles, but also to put pressure to help to bring blood back up to the heart. Well, it also had a very thickened band-like region, and that thickened band-like region is that iliotibial band. So this iliotibial band on the outer side, one of the structures you're responsible for, is also a part of the origin, a part of the insertion of our gluteus maximus. So notice it's not in your list if you don't put it on. If I ask you for the insertion of the gluteus maximus, you don't have to list it, but I do want to point it out because it is something that is important to see. Anytime we have a gluteus maximus or a maximus anything, we're going to have a minimus something too. And sure enough, we have a gluteus minimus, but it's a deep muscle, but it's not on your list, so you're not responsible for it. What you are responsible for, though, is the one in between. There's also not only a gluteus maximus and a gluteus minimus, but there's a gluteus medius. And again, don't get confused by the name. It is medius by name because it is medium in size. Medius does not mean medial. In fact, it's absolutely the opposite. While the gluteus maximus is a posterior muscle, your gluteus medius is actually a lateral muscle. And we really see that when we look at the origins and insertions of it. When we look at the origin and insertion of it, notice that the origin of the gluteus medius is basically the fossa of the ilium, the lateral fossa of the ilium. Notice it does not go up onto the crest. It does not go up onto the crest. It's basically below the crest is where it is. So it has a very lateral origin, and it inserts into the lateralis most feature of the hip, which I'm sure you remember from your skeletal system exam and study guide, that review. The lateralis most point of the hip is the greater trochanter. So basically, if we were to look at it from the side, and again, you're not having to draw it on this, but let's go ahead and put it on here in green just to emphasize it. Notice basically it starts here from the outside and goes to that lateral end. So it really is a lateral muscle. Notice we also see that with their actions as well. The gluteus maximus brings the leg back, right, extends the hip, whereas the gluteus medius abducts the leg, brings the leg out. Lateral fossa of the ilium for the origin, that would totally be acceptable, yes. That the lateral fossa would absolutely be acceptable. All right. Now, so that is the muscle. The other thing that I wanted to point out about this muscle is this muscle, from a clinical standpoint, is important because this is an important muscle for injections. Right? In the movie, it's always funny when they get the injection right in the butt because it's more humorous from a movie standpoint, but it isn't practical. When you are looking for a muscle for an intramuscular injection, there are basically three things that you are looking for. First, the muscle has to have enough mass where it is able to receive the medicine. And the gluteus maximus and the gluteus medius would both do that. The second thing that we're concerned about is the blood flow, right? How well vascularized is the muscle? If it's too well vascularized, that medicine will be distributed through the body too rapidly. If the blood flow is too slow to that muscle, then the medicine congests in that muscle and doesn't circulate quickly enough. And again, both the gluteus medius and the gluteus maximus have a similar blood flow to them as well. But the third consideration is innervation. You want to watch out for large nerves. And for the gluteus maximus, there is a large nerve that comes out here on the back, right? Comes right out of this opening right here, coming out here and down the leg. In fact, it's the largest nerve in the body, the sciatic nerve. And so a injection into the gluteus maximus runs the risk of hitting that nerve and causing damage. So when we do a intramuscular injection, it is much more common in the gluteus medius than actually the gluteus maximus. Basically what you do is with your fingers, you find the iliac crest, then you go about 2 inches or 2 fingers down, and then boom, you make the injection there. In fact, it is one of, I don't know what I'm doing here. Stop that. two most common muscles for injection. One is the gluteus medius. Anyone know what the second most common muscle for an intramuscular injection is? Let me rephrase that. I know you know. Who can tell me? What other muscle do you get your intramuscular injections into? Yeah, the deltoid, absolutely. Which notice also happens to be, now be careful, thigh is more kids. Thigh is kids because, again, we want that mass. It goes back to the mass. In adults, and again, it does vary, certain medicines, certain boluses, but the most common locations for adults is going to be the deltoid and going to be the gluteus medius. All right. So that's something I wanted to say about those to distinguish the difference between those. Let's talk a little about the hamstring muscles. First, why the heck are they called the hamstring muscles? That's always a good question. And the answer for that is actually really interesting. Notice down here, and I'll see what I can do about filling that in. Excellent. Oops, no, I didn't want to do that. Notice this down here. Get rid of that. In the back of the knee is this diamond-shaped region. This diamond-shaped region is what is known as the popliteal fossa. Our leg anatomy is actually similar to the leg anatomy of a pig. And when you are butchering a pig, one of the things that you want to do is to hang and cure the leg to make your ham. And so what they do is they take a large meat hook, stick it into this popliteal fossa, into the large meat of the leg, and that is how they hang it to cure that meat to produce a ham. And so that's why this is called the hamstring, because this is where you string up the ham. Right? As I said at the beginning of this section, right? If you want to have the philosophical debate about whether or not meat is murder, we can have that philosophical discussion if you want. But the one thing that we cannot discuss, because it is 100% true, is meat is muscle. And so absolutely, this muscle anatomy is similar to that of the ham, of the pig, and that's where it gets its name. Notice this is comprised of 3 muscles, but notice also we have 4 origins because we have a biceps. And with that bicep, we have 2 bellies. Now notice this is the bicep femoris, the bicep in the femoral region, as opposed to the bicep brachia, the first one we learned. And while their names are similar, their anatomy is not. Notice when we look at it, we really only see one of the bellies of the bicep femoris in this view. Whereas if you remember, the 2 bellies for the bicep brachia were side by side. Here, what we're seeing is the long head of the bicep brachia, and deep beneath it, so directly underneath it, basically, let me cheat and grab my highlighter again. Basically here underneath it, directly underneath it, would be where you see the short head of the bicep femoris. Oh, do I still have? I don't. Hold on, let's go back to, because I know it is good here. Let's do that again. That was fun. Here is our virtual anatomy lab. In our virtual anatomy lab, if we go to our limbs, there is our arm. Here's a leg. Notice here we are seeing the superior part of the hamstring. What I want to show you, and I think our models show it nicer, so let me bring my annotation tools up again so I can highlight them. Highlighter, green. Again, this is the long head of the bicep femoris. But notice on the opposite side, on the medial side of the leg, we have 2 muscles that also are in a similar arrangement. Notice the long head is on top of the short head. Over here, we have, and let me change colors, our semitendinosus, which is a superficial, small, thin muscle right here That is our semitendinosus. And deep to that, let's see what black looks like. Notice there is a little bit of a muscle sticking out on the side there, and a little bit of the muscle sticking out on that side there. That deep muscle that we see beneath it is the semitendinosus. Pardon me, semimembranosus. So semimembranosus is the deep one. It is wider, and the way I remember it is the membranosus is more medial. Notice because it's wider, it is more medial, whereas the semitendinosus is more superficial and more on the top. And I think there was a question. Linea aspera. Remember, the linea aspera is on our femur. Remember, on our femur, there's that raised ridge that starts on the back of the femur, and then it gets a little fuzzy up near the top. And then remember, it is from that linea aspera that the gluteal tuberosity comes off of. So if you remember the back of the femur, there are these raised ridges, the linea aspera, and then the gluteal tuberosity. And so that's what those are again. All right, let's go back here. So again, we've got those three muscles here of the hamstring, but if I remember correctly, oh, and there we see, oh, great view of the semitendinosus. So again, notice the semimembranosus. Semimembranosus is the deep one beneath the semitendinosus. Again, notice here's that long head, and here finally we see a little bit of the short head coming out, but notice, oh, I swear they have it. They don't. Huh. Okay. Well, then let's go the other place that I would be. My lab of mastering. Again, I know how you get to your study area is slightly different from but once you're here, it's going to be the same. Here in your lab tools, go to your practice anatomy lab. That's what the P stands for. I always get that wrong. Practice anatomy lab. Launch that. Let's do two things first. Let's first look at the animation. Lower limb, bicep femoris.
Biceps femoris is the lateral member of the muscle group located in the posterior thigh, known as the hamstrings. Biceps femoris has two heads, the long head and the short head. The origin of the long head is by a common tendon with the semitendinosus from the superior and medial surfaces of the ischial tuberosity.
And again, all you need is
ischial
ischial tuberosity.
The short head are from the lateral lip of the linea aspera and the lateral supracondylar line of the femur.
Again, just linea aspera.
The fibers run inferiorly and laterally to insert primarily onto the lateral surface of the fibular head, but it also inserts on the adjacent surface of the lateral condyle of the tibia. Both heads of the biceps femoris flex the leg at the knee and laterally rotate the knee during extension from the flexed position. The long head also extends the thigh at the hip. The long head is innervated by the tibial nerve. The short head is innervated by the common fibular nerve.
Now, notice that rotation of the knee is not one of your actions you're responsible for this, because really it's not so much of a rotation as much as it's more of a stabilization. It stabilizes the knee as opposed to actually moving and twisting the knee. So that is why it is not considered part of your list. The other place I wanted to look at this, though, is I wanted to go to the muscular system, to the lower limb, because, and then if we use the show the gallery, I can get to what I want. Perfect. Let's start here first. You guys can see this okay? Yes? Again, when I'm doing things like this, I'm always concerned about what you guys can see and what you guys cannot. Okay, excellent. So notice here we have the long head of the bicep femoris, and the short head would be just peeking out beneath it. Here, again, we have the semitendinosus, that is the superficial, on top of the, again, notice wider, and so therefore more medial semimembranosus. But notice if we go to the next picture, what they've done is they've removed it. And so notice when they've removed those superficial muscles, what we can now see is that bicep femoris coming off of the linea aspera and coming down and wrapping around. So that bicep femoris has to be, you have to remove the long head for us to be able to see the short head. And then here we see some of the other muscles we're responsible for us now. But again, what I wanted to show is primarily that short head of the bicep femoris. You really need to remove the long head to be able to see it, so because it is a deeper muscle. All right. Questions on that? So notice it is technically 3 muscles here with the hamstring. But if you think about it, there's a tremendous amount of symmetry. Our semimembranosus and semitendinosus, we have a superficial muscle on top of a deep muscle. And our long head and our short head of the bicep femoris is a superficial belly on top of a deep belly. So they have similar orientations that way. All right. Notice again, these are part of a muscle group, which means they share at least one attachment point. And notice for for this muscle group, that shared attachment point is the ischial tuberosity, the long head of the bicep femoris, and the semimembranosus and semitendinosus all originate there. But notice the semitendinosus and semimembranosus both have the same insertion. So the semimembranosus and semitendinosus have the same origin and the same insertion. They're slightly set off from each other, but not by much. They all have it. In fact, if we look at, and I think we can go back here. Did I close the, no, that's what I want. That's not the view I want. That's not the view I want. None of these have the view I want. All right, then let's go to here. All right, none of these things are truly giving me what I want, but that's okay. What I will say is, and that we can see it here, is this one shows me what I want. As we'll talk about, there is not 1, not 2, not 3, but actually 4 muscles that all are going to insert into the medial condyle. My spotlight. Why are you not giving me my spotlight? Am I not sharing the right thing anymore? Oh, that's why. That's why. There we go. Not 1, not 2, not 3, but 4 muscles that are all going to insert into and underneath the medial condyle of the tibia. So if we look at the pretty picture from our bones, right, this spot right here, under the medial condyle of the tibia, there are 4 muscles that all insert into. And the first 2 are the semimembranosus and the semitendinosus. So if this bone is shown and I give you this spot, you know there's going to be 4 muscles you're going to be responsible for identifying. And those are going to be, the first 2 are the semimembranosus and the semitendinosus. likely I'll be curving the exam. Again, like I said, this is the first time I've done an exam like this, so I'm going to see the scores before we decide whether they need to be curved or not. So I can't say for certain one way or another, but hopefully you guys will do well enough where I don't have to curve it. Wouldn't that be fun? All righty. So we did that. And so again, what I was finishing with, semimembranosus, semitendinosus, both same origin, both same insertion. So not surprisingly, they both have the exact same action. The last thing I wanted to talk about, actually that's not true. The next thing I want to talk about is our next muscle group, and that is the quadriceps femoris, where we have 4 big muscles here. And again, this is considered an anterior muscle group, right? But in reality, the vastus lateralis is really on the lateral aspect of our leg. In fact, when you look at its origin, you see some of its origins are on the anterior side, some of its origin is on the posterior side. The same thing for the vastus medialis. It is really a medial muscle with some of its origin on the anterior of the leg, some of its origin on the posterior of the leg. Then we have this orientation we are growing familiar with. We have a long straight muscle. That's what a rectus is a long straight parallel muscle in the femoral region. So that straight rectus muscle, the rectus femoris, which is a superficial muscle, and deep underneath it is the vastus intermedius. These 4 muscles, so basically the rectus femoris and the 3 vastus brothers, are the quadriceps femoris. Now, like I said, it's considered an anterior muscle group, even though the vastus medialis is medial and the vastus lateralis is lateral. And the reason for that is its insertion, and more specifically, its complete insertion pathway. It is a complete insertion pathway, right? Up to this point, when we've talked about insertions, we've identified insertions, and all 4 of these muscles indeed have, so let's say it this way, all 4 muscles have the same insertion, and that insertion is the tibial tuberosity. That bump we've talked about, that is on the front of your tibia, right underneath your kneecap. And that is the same for them. However, what makes these anterior muscles is their complete or entire insertion pathway. All of these 4 muscles, and these 4 muscles will be the only muscles where I'll ask for an insertion pathway. So being the smart, sophisticated students that you are, if you see a question that says describe or identify the complete insertion pathway, then you don't even have to look at the picture. You know what the answer is going to be, because it is going to be one of these 4 muscles. And that entire insertion pathway starts with the quadriceps tendon. So all of these come together into a single tendon. That quadriceps tendon then attaches to the kneecap, the patella. From that patella, we then have a dense regular connective tissue that connects bone to bone, and that is our patella ligament. One of the things we learned on our bones when we learned the knee, patella ligament. And that patella ligament then feeds into the tibial. So if I just ask you for the insertion of any of these 4 muscles, you just have to say tibial tuberosity. Right? If I just say insertion, all I want is tibial tuberosity. If, on the other hand, I say the insertion pathway, then I want these 4 things: quadriceps tendon, patella ligament, tibial tuberosity. No, all 4. If I ask for the insertion pathway, the entire insertion pathway, you must give me all 4 of these. So yeah, if I ask for the entire insertion pathway and you just give me one of these things, patella, that's not an insertion pathway. A pathway is a path, and this path has 4 steps to it. So you have to give me all 4 steps. Now, again, if I just ask for the insertion, then all I want is the last step. Where does it actually attach to the bone? Tibial tuberosity. But if I want the pathway, and again, just like be specific, this is a special term that should tell you there's something significant and important about it. And that is, tells you exactly what the answer is. That's the advantage of that. All righty. I think the rest of these are pretty simple and straightforward, so I don't need to talk anymore about their origins, insertions, or actions, or any of those things. I think it's pretty simple and straightforward. But I did want to talk about this insertion pathway. The other thing that I want to point out while we are here are these other 2 muscles. Well, actually, 3 muscles we'll talk about here, but in particular, these 2 muscles, the sartorius and the gracilis. I'm not sure you mean by should be able to identify items. Are those the things at the bottom? Structures. So remember, those are things that I mentioned are structures. Oh, and here are some of the good ones, right? We've talked about this. This is a good picture that shows some of these. Let's go ahead and make these green so we can identify these pretty nicely. Yes, and a calcaneal ligament and Achilles ligament, calcaneal tendon and Achilles tendon. It's not calcaneal ligament. Calcaneal tendon and Achilles tendon are both the same, and I would accept that. Here is that inguinal ligament. Remember, the inguinal ligament we saw was an attachment point for our, I know that's what you meant. And you see me type. I type wrong all the time. Not a big deal. It is that ligament that's part of the attachment point for our abdominal muscles. Remember, it's a ligament, so it goes bone to bone from the superior anterior iliac spine to the ischial tuberosity. We talked about the iliotibial band is one of the structures you're responsible for. And notice this great picture from your textbook actually draws us a little bit of that fascia lata that wraps around it as well. Know what insertions? The insertion, and you need to know the insertions of all the things that are on the list. The calcaneal tendon is a tendon for a muscle, so it doesn't have its own insertion. It basically attaches to the insertion of the soleus and the gastrocnemius. It's how the gastrocnemius and the soleus get to the calcaneus. So tendons themselves don't have insertions; just muscles have insertions, unless you meant a different insertion. Oh, no, features. And again, I know it says features, but remember the key word I will use on the exam for any of those things is structures. Identify the structures. These structures, and that's my strong and standard way of telling you, these are not muscles. These are things that are not muscles. And again, here we see 3 of them, 3 of them right here. The fascia lata, the iliotibial band, and the inguinal ligament. So those are some of those structures that you could be responsible for. Great questions. More? All right, let's talk about these next 2 muscles then, as I was talking about. The gracilis. Oops, no, that's not what I want. We've done the trunk ones already, so I don't necessarily want to go back to that. What I would encourage you to do is to go back and listen to the lecture where we talked about them. If there's a specific trunk muscle you have a question about, I'd be happy to answer that question when we have a review, but I don't have the time to go over all of them, so let's go through the leg first. And then, like I said, if you have a specific one, I'll answer that, or I would encourage you to go back to the lecture where we talked about it. So the next 2 muscles I want to talk about are the gracilis and the sartorius. Notice the sartorius. The sartorius is actually the longest muscle in your body. But notice it is a narrow strap-like muscle. There's not a tremendous amount of fascicles in it. So it's certainly, while it is the longest muscle, is certainly not the strongest muscle. But it is technically the longest muscle. It starts at the highest point it can find, and that is that superior anterior iliac spine. And it is the third muscle that inserts into the medial condyle of the tibia. Notice it crosses both the hip and the knee, so it affects both. And this is that muscle that allows you to cross your leg like a boy would cross your leg. You flex your knee, you flex your hip, you abduct your leg, and you laterally rotate it. And that puts your ankle on your other knee. Right? Notice it starts lateral and it comes medial. We have another superficial muscle that is the fourth muscle that inserts into the medial condyle of the tibia. And while this one inserts medially, it also starts medially. And that muscle right there is the gracilis. Oops. The gracilis is our medialmost muscle. We see that with its origin, it originates at the medialist most point, basically right at the inferior angle of the pubis, and again goes into the medial. Notice, like the sartorius, it crosses both joints, the hip and the knee, so it is going to flex both of them, right? But being medial, it actually medially rotates and adducts. And I had a student a few years ago who, again, remember I told you how in class normally I make you guys stand up and demonstrate. Not only did she demonstrate, but she took a picture for me so that I could share it in my lectures. She said the gracilis is what allows you to do what she called her fangirl pose. So as we see here, we'll come to that. There we go. You can see here is her fangirl pose. With her fangirl pose, I assume she just got tickets to a Justin Bieber concert or something, but you can see she is flexing her hip, she is flexing her knee, she is adducting it, and she is medially rotating. So with that action, she's getting that fangirl pose, and that is what that gracilis allows you to do. All right? So those are two similar muscles. They're both superficial, but the big difference is that sartorius starts lateral and comes medial, whereas the gracilis starts medial and stays medial. But both are superficial. Right? The other big one that I wanted to talk about, because I think there is a bit of confusion sometime on this, is again another muscle group. This muscle group is what is known as the iliopsoas. Right? Notice the sartorius, the gracilis, even the rectus femoris. The rectus femoris, being a parallel muscle, is one of the, actually, pardon me, it's not parallel. The rectus femoris is a bipinnate muscle. So it has that single tendon that goes down the center, and it has fascicles that come into it from both sides, making it one of the strongest muscles of the body. And that makes sense because our leg is a big, huge mass of meat that needs to be moved. But the other major mover of the leg, the other major flexor of the leg, is actually a muscle that actually starts inside your abdominal pelvic cavity. Inside your abdominal pelvic cavity, anchored to the lumbar vertebrae, anchored to the inside of your pelvis, are three muscles, really two and a half. One of those muscles is the iliacus. That's the one on the inside of the ilium. The other is the psoas major, which is the big wide muscle that attaches. And I'd say somewhere close to 60% of people also have this thin, small muscle known as the psoas major. But again, these are not muscles that are on your individual lists, so you don't need to know them individually. You just need to know them collectively. And collectively, these muscles are the iliopsoas. They have a big, huge, strong base on your pelvis, on your lumbar vertebrae, and basically attached to the front of the leg so that it can flex the leg. So these two muscles, the rectus femoris and the iliopsoas, are the two big major flexors of your leg to move that big, huge, massive piece of meat through space. All right. So we saw that. Oh, one more. This great, I love this muscle. It's always the tiny muscles that have to compensate by having the big, huge, massive names. Tensor fascia lata, right? This muscle has a big, huge, massive name, but again, the name tells you everything about it. It is a tensor of the fascia lata. The fascia lata, remember, is that sheath that wraps around the leg, and this muscle, through the iliotibial band, puts tension on it. So this is basically the cuff that helps to put tension on that fascia to help to, again, stabilize the muscles and lead to the circulation. Of course, it's an anterior lateral muscle, so it is also going to play a role in helping to flex and abduct the leg. But notice that tiny, swinging the leg out to the side is not going to be its primary function. One of the cool things, one of the things I like about this is on our bodybuilders, sometimes this muscle, when they flex it right, it gets huge and massive and really obvious and easy to see. So it's kind of nice that way. And that is one of the cool things about these muscles, right? When we look at these legs of our bodybuilders, they are pretty crazy to look at. Remember, one of the things we need to be able to do is identify these on our bodybuilders. But there's a lot. One of the great things about this is we have some great definition, but there are some changes that take place, right? One of the changes that takes place, and is one of the less obvious ones, is what happens to the rectus femoris. Notice the rectus femoris normally goes the entire length of the leg, all the way to the kneecap. But in fact, what happens is this right here is the rectus femoris. Notice we can clearly see the actual, here, let me draw it, the tendon in the center of it. So we can actually see that tendon where the fascicles are all coming in on the sides so that we can have it. And this is what happens with this muscle. This muscle, because it has a long tendon, can't get longer. Notice it definitely gets wider, definitely gets bigger, but the vastus medialis, the vastus lateralis, and the vastus intermedius underneath it all get big and massive and basically push it up. And because of its bipinnate shape, it can't really grow longer. So as it gets pushed up bigger, it moves further and further away from the knee. Notice we can pretty nicely, let me see if I can, that does it. Here we can see our vastus medialis. One of the nice things about the vastus medialis is always going to be right next to the knee. It always makes it easy to find. And then we can distinguish that from the vastus lateralis, which is this one here. And remember, our vastus intermedius is deep underneath. No matter how big that vastus intermedius gets, we're never going to see it. Notice also, we can actually see the stripe-like muscle that is that sartorius. Notice we can see the stripes of that sartorius starting lateral and wrapping around the medial head of the vastus medialis to come in from the side. But notice we don't see the gracilis or the abductor magnus. Even as big as these people get, right? Again, if you think about adducting the leg, which is what these adductor magnus and the gracilis do, gravity pretty much does that for us, right? We don't really use those muscles a lot, so it's really hard to get tension in them. There are those torture device contraptions at a gym that you can get into where you can strap your legs into them and be able to basically pull it together, right? It's that machine that spreads you all out where you can't make eye contact with anybody in the gym while you're using it, right? To kind of tone the inside of your leg. But obviously nobody wants to spend a lot of time on that. So notice even on our bodybuilder, there's not a tremendous amount of tone in those, and we can't distinguish those muscles. But these four we can clearly see on the thigh of our bodybuilder. And in fact, one more. Notice his pesky pants are in the way, but notice you can see a teeny bit of that tensor fascia lata starting to, I'm going to go ahead and erase that one so it's easier to see. I'm going to get this one as well. Notice there's a little bit of a raised portion where you can actually see a little bit of that muscle starting to come out. So if we had a different view and if he had a higher cut to his shorts, we might actually be able to see that tensor fascia lata as that distinct muscle that can stand out on the anterior and lateral aspect of the leg. So again, when we look at these, it doesn't look real when you look at some of these bodybuilder legs. But when you look closely, they actually have tremendous definition for many of the muscles, where we can see the sartorius and the vastus medialis and vastus lateralis and rectus femoris, all muscles you are responsible for on our bodybuilder. All right. This, with a deeper view, we get a nicer look at that gracilis, which is that a medial muscle starts medial, goes medial, right? And then notice also, oops, we have our adductor magnus here. Notice the adductor magnus is part of an adductor group that includes the adductor longus and the adductor brevis. You are only responsible for the adductor magnus. Notice it is deep to the gracilis and a little bit anterior. So that's how you're going to recognize it on the models. It is deep and anterior to the gracilis. The reason I make a point of emphasizing this one is one of the things back in ancient times when we had sports, you would hear about basketball players or football players or even sometimes baseball players who would pull a groin muscle, right? That's a common term that they use in sports for an injury where you pull a groin muscle. Where typically the muscle is being injured, probably in about 70 to 80% of those quote unquote groin injuries, it typically involves an injury of this adductor magnus muscle. And like I said, one of the reasons for that is it's hard to put tension on it. It's hard to get tone in it because being an adductor, its primary job is to bring the leg down. And when you bring the leg down, again, gravity does it, so we don't put a lot of tone. No, you will not be responsible. Even though the gracilis is a superficial muscle, you will not be responsible for it on the bodybuilder. Because remember, with the lack of tone means we can't show it. We can infer where it is. We can kind of guess where it would be if we go back to our bodybuilder, right? It's probably, it's the medial most muscles. So it's probably this may be a little bit there, and this may be the abductor magnus, and this may be the adductor longus. But again, the tone here is very different from the tone here. So I'm not going to hold you responsible for those medial muscles on our bodybuilder because they just typically don't have the tone in them. All righty. Oh, it's not that it wasn't the gracilis. The gracilis is there. He definitely has a gracilis, but we're not able to see. If you notice here, there's also the abductor magnus. There's a little bit of the adductor brevis that comes out, the abductor longus. We just can't distinguish the individual muscles. They're all definitely there. We just can't tell them apart. So that's why you wouldn't have to, I'm not going to just point at an area and say what muscle should be here. It's going to be one that's going to be clearly defined. All right. Last two muscles that I wanted to talk about is down here in the leg, starting first with the gastrocnemius. There is an adductor group. There are the adductor groups made up of the adductor magnus, brevis, and longus. So that is a muscle group on the inner thigh. The gracilis is not a part of that muscle group. And more importantly, you don't need to know the adductor group. You just need to know one of the muscles of the adductor group. So the same way like for the levator ani, you just need to know the name of the muscle group and not the individual muscles. This is the opposite here. You don't need to know the muscle group. You just need to know this one muscle, the adductor magnus. Okay, does that make sense? But yes, technically there is a muscle group here. You just don't have to know. There are a lot of muscle groups. You just haven't had to know. So limited number. Like I said, there are over 600 skeletal muscles in the body. So there's so many muscles, so many muscle groups, so many things we haven't talked about. This, I know, as intimidating as this test seems, it really is the tip of the iceberg. There's so much more. Now, the bicep brachia and the bicep femoris have similar names, but as we saw, their anatomy is not similar. The gastrocnemius's name is nothing like bicep femoris, but its anatomy is much more similar. Notice with the gastrocnemius, like the bicep femoris, we have two bellies side by side, right? One is medial, one is lateral, so we have a medial one over here. We have a lateral on. Ooh, here we go. Here we see, this is nice. Right? 1, 2, 3, 4. There are those 4 tendons we talked about that are all coming down into the medial condyle of the tibia. So we've got again, the sartorius, the gracilis, the semimembranosus, and the semitendinosus. All 4 of those come in. All right, but that's not what I wanted to show you. So here we have, again, the medial belly and the lateral belly side by side. Prominent superficial muscles that are over the lower leg, just like we have prominent superficial muscles that are the bicep brachia. Remember also, the bicep brachia sits on the humerus but doesn't actually attach to it. And the same thing is true here with the gastrocnemius. It sits on the tibia and fibula, but it actually originates from the femur, the medial and lateral condyles of the femur up here. And as was already mentioned, oops, wrong color. It inserts into the calcaneus, our heel bone. And of course, as you guys pointed out, it inserts via a specialized tendon, and that specialized tendon is the calcaneal tendon, or as I already mentioned, what you can call the Achilles tendon. Crossing 2 joints, knee and ankle. And one of the important things to remember at the ankle is if it is attaching behind the ankle, the medial and lateral malleoli, then it is going to basically plantar flex. And if it attaches in front, it dorsiflexes. So this one points your toe and flexes your knee, right? Like that true love kiss that we talked about. Well, not talked about, but was on your handout that was due today. There is one other way our gastrocnemius is like the bicep brachia. And that is, while it is the big prominent muscle that sits on top, it's the superficial ones. We talked about everybody, all the boys wanting to do the curls for the girls so they can have big biceps. Well, this is what we commonly think of as the calf muscle. And if you're not happy with your calf muscles, they actually have calf implants that you can put in to make your calves look more impressive. All right. Many, many years ago, he's still very famous. He's now famous for different things. He spends a tremendous amount on Twitter. But there was a very famous wide receiver for the Cincinnati Bengals by the name of Chad Johnson, whose number was 85, and so actually changed his name legally to Ocho Cinco for a while. One of those very character wide receivers, but amazing legs. He once raced a horse and beat it in a race. Huge, powerful, fast individual with amazing calf muscles. A company actually paid him $500,000 to make a mold of his gastrocnemius so that they could use it to make implants. So that if you're not happy with the way your calves look, you could get calf muscles like Chad Ocho Cinco Johnson. And they paid him $500,000 to do it. So it's this big, superficial muscle that stands out that everybody loves and everybody is impressed by, just like the bicep brachia. But just like the bicep brachia, there is a muscle underneath that helps. And the same thing is true here. Here, deep to our gastrocnemius, and notice we don't necessarily see it here, but notice here where the gastrocnemius is cut, we see that there's this big flat muscle that is folded into the space underneath the gastrocnemius. And that's actually where it gets its name. Soleus gets its name from the sole fish, which is a broad, flat fish that lays on the bottom of the lake or ocean. I don't know if it's saltwater or freshwater, but it's a big, flat fish. And this is a big, flat muscle, this soleus. But notice we see a little bit of it peeking out here from the back. And notice also its insertion is the same. It also attaches to that Achilles tendon and attaches to the calcaneus. All right, so that again is just another way that the gastrocnemius is similar to the bicep brachia. I like that. That's actually a good essay question. All right, I didn't use that, but maybe next year. All righty. Those are the main things. Again, there are still plenty of other muscles on here we haven't talked about. Oh, here's another great view. Here's where I wanted to see it. Look at this. Here we see it. Excellent. So again, notice, oh, let's use lines. Great question. I'll answer that in a second. 1, 2, 3, 4. So notice there are 4 different tendons that all come onto the medial aspect of the condyle of the tibia. So again, it's the gracilis, sartorius, semimembranosus, and semitendinosus all come into that. Oh, one other one that I want to point out, then I'll answer your question in just a second. But I want to say this before I forget. Notice one more muscle I want to talk about is the extensor digitorum longus. Notice this is a muscle whose tendon comes out and splits and goes to the digits of the toes, just like we had in the forearm. All right? So there is an extensor digitorum in the forearm, but here we have an extensor digitorum longus, and that is the difference. One is just an extensor digitorum, one is the extensor digitorum longus. The longus is in the leg, and again, that's one way to remember, longest leg, right, whereas just the extensor digitorum doesn't. I remembered actually the longest leg is clever. It obviously has longer tendons to get all the way to the toes, so that's how I remember. The longer tendon to get to the toes, extensor digitorum longus. All right, now let's go back. That's a great question. So another way, oh, wrong direction. Clear. Another way that the gastrocnemius and the bicep brachia are similar is that they both cross 2 joints. They both cross the knee. They both cross, well, this one crosses the knee and the ankle, whereas the bicep brachia crosses both the shoulder and the elbow, so it affects both. But remember, the brachialis, which sits underneath the bicep brachia, starts on the humerus and attaches to the forearm. So it only affects the elbow. Same thing here. Our gastrocnemius crosses 2 joints. Our soleus attaches to the tibia and the fibula. It's got this U-shaped origin to it, and then it just inserts into the calcaneus. So it only crosses the ankle. So whereas the gastrocnemius flexes the knee and plantar flexes the foot, the soleus only plantar flexes the foot. Right, so that's why there's the differences, even though they're in very similar location, there's a difference in their actions, and that's because of the difference of their origins. The gastrocnemius crosses 2 joints, the soleus only crosses 1. All righty, excellent. With that, I think that, like I said, that isn't all the muscles, but I think that's all the tricky stuff that I wanted to make sure you are aware of going through all of this to help you to prepare for the exam for that. All right. So like I said, that is everything that you guys are going to be responsible for on the exam that, again, that are tricky. Obviously, there's still more material you're responsible for. So they please explain number 6 on the first page of the skeletal muscle study guide. I don't have the skeletal muscle study guide in front of me. What's number 6? Can someone tell me what the question is? Again, you have to tell me what the question is. So I don't know the buccinator. What? So is it? Did I give you a muscle and you have to come up with an action, or did I give you an action you have to come up with a muscle? Is that the thing you're talking about? Oh, so you just want to understand what the buccinator is. Is that what you're asking me? Oh, that I can do. Here, let's go back to, did I close it already? I did. Right. Okay. So what you're talking about is the study guide and the 6 month muscle, the buccinator, right? Although it doesn't, it's not bold, so you don't have to. You don't have to. Wait. What? Number 6 on the first page of the skeletal. Oh, that one. Okay. Oh, got it. Got it. Okay. Someone confused me with the buccinator. Um. All right. I get what you're saying. So what I was doing there, and let's we can go here to figure this out. This is as good a spot as any. Can you guys see that? Yeah, okay, perfect. So one of the things that I wanted to remind you of, and again, it's just a different way to study this material, is to think in terms of organization. You are responsible for these bones. So when we think about, let's do this, let's increase, no, don't do that. Increase the size again, scroll down. There we go. And annotation tools. So, yeah, okay, I understand now. I got it now. So, again, I'm trying to think to give you ways to think about and master this material. So, one of the ways to do that is in organizing them, right, and thinking about how these things relate to each other. Think about it when we learned the bones. We learned about each individual bone and bone feature, but then we put them together into the skeleton to see like what holds our pants up, or what is the widest point of the shoulder, or something like that, to see how these things relate to each other. This is another way to think about how these things relate to each other. So notice there are 11 muscles that either insert or, and let's see how small I can make this, insert or originate from the tibia. So one of the things you could think of is to try to determine, if you looked at a tibia, what those 4 things would be, or what those 11 things would be, right? For instance, 4 of them are going to be all of the muscles of the quadricep femoris. All of the muscles of the quadricep femoris all attached to the tibial tuberosity. So those are 4 muscles right there. I just finished telling you that there are 4 muscles that insert into the medial condyle of the tibia. And let's go back. The 4 muscles here are the rectus femoris, the vastus medialis, vastus lateralis, and vastus intermedius. We mentioned there are 4 muscles that, oops, that didn't work. Move that a little bit. 4 muscles that basically insert into the medial condyle of the tibia. And as we talked about, they're the gracilis, the sartorius, the semimembranosus, and the semitendinosus. Boom. Just that simply, we have identified 4 of them. We didn't talk about it, but again, just the bold muscles that you're responsible for. One of them is the tibialis anterior, right? This one over here. It basically inserts from the lateral surface on the front over here. Notice there's no room for it over here medially because there's 4 muscles that insert here. There's 4 muscles that are inserting into the tibial tuberosity, but right over here, laterally out on the side here, is where the tibialis anterior originates. And then that tibialis anterior comes down and around the leg. There's that there. So that is 9. Then we need 2 more. One of those, if we look at the back side, We know out here, we have the soleus, as we've talked about, has this kind of U shape. And let's go ahead and draw this. This kind of U, oh, not red, not blue, red. The soleus kind of has this U-shaped origin on the back of the tibia, so that is it there. And then remember how we've got our semimembranosus and semitendinosus that come over here on the medial condyle. Remember also the two heads of the bicep femoris, two heads, but really only one muscle. Insert into the lateral condyle and the head of the, oops, that does not show up. So we'll cheat and move it right there. Again, into the blue, it inserts into the lateral condyle and the head of the tibia over here on the lateral side. So notice with that, those are 11 muscles, bold muscles only. Obviously, the fibularis, the peroneus longus or fibularis longus is going to be there. There are other muscles, the extensor digitorum longus. There are plenty of other muscles that attach to the tibia, but they're not bold ones. They're not ones you're responsible for the origins and insertions. So of the muscles that you are responsible for the origins and insertions, notice 11 of them attach to the tibia. All right? 9 of them attached to the os coccyx, 7 of them attached to the scapula, and so on and so forth. So it's just the reason I have that there is just to give you another way of thinking about this material to help you to synthesize the information so you can understand the relationship of these muscles to each other and to the bones. All right, I apologize for my confusion and not understanding the question, but did I at least give you the answer that made sense? Excellent, awesome. All right. So we answered that question. Next question about the platysma. What's your question about the platysma? Ashley? No, it has. It really has two actions. The platysma basically is this big drape-like muscle that comes up over your jaw. So basically, it has two actions. The first is to depress the mandible, bring it down. The second is because it comes up over the lip, the chin, it attaches to the lip, so it gives you the pouty face. So that pouty face, that ability to extend the lower lip is from that. Remember, we've talked about different ways that you can see these muscles. Notice if you clench your jaw so that your jaw doesn't move down and you contract this muscle, and I don't know, this is going to probably look horrible on camera, but notice you can kind of get that angry Hulk neck. The angry Hulk neck. When you get that angry Hulk neck, that occurs because that's actually that big drape like platysma. So it's primarily about bringing things down, right, as opposed to so much back and forth. I guess technically, if you were able to contract one, it might maybe pull the head a little bit to the side, but no, whipping the head around is not really one of the actions of it. And certainly, even if it is capable of a little bit, most importantly, it's not on your list, so you're not responsible for it. You're only responsible for 2 of its actions if it has more, and those are depressing the mandible and protruding the lip. But really, I don't think of the platysma as being something that can whip the head around. I feel like there was another question someone had. Oh, the trunk muscles. Was there a particular trunk muscle that there was a question about? Who had the trunk? I can't find the question. Oh, the structures of the trunk. That's what you wanted, the structures of the trunk? That we can do. Perfect. Let's switch back to the lecture. Oh, you guys were talking about the buccinator. Buccinator is that deep muscle. Notice between the zygomaticus and the masseter, find the masseter, find the zygomaticus. That's the one that squeezes the lips, right? Not protruding it, we're not protruding the lips, right? We're squeezing the cheeks. And my favorite example is, of course, the greatest movie of all time is, of course, Zoolander. And notice with Zoolander, notice he's maybe protruding his tips a little bit, but what he's really doing is squeezing those cheeks in. And squeezing those cheeks in is that function of that platysma. So there we've got that. But what we want to get to is these structures. Here we go. Excellent. So I believe there's 1, 2, 3, 4 structures for the trunk that we are responsible for. The first one is the big, huge, flat tendon that is here. And this big, huge, flat tendon is the tendon of the external oblique. So we have the aponeurosis, because remember that's the term for a big, broad, flat tendon. That is the aponeurosis of the external oblique. Remember, we looked at it from this point of view. Notice when we looked at it from this point of view, we see here again that big, flat tendon that comes across. But notice we also see the internal oblique and the transversus abdominis also have aponeuroses. You're just not going to see them from the front because this is the most superficial one, right? This is the most superficial one. However, they do come together in the middle where they mix with the fibers, the tendons of the other side, forming that line straight down the middle between the rectus abdominis, and that is the linea aspera. As we talked about, the inguinal ligament goes from the superior anterior iliac spine to the pubic tubercle. It is part of what holds these abdominal muscles in place. And as we also talked about, this is where males can get that inguinal ligament because there is a passageway, a canal through it that the spermatocord comes through to go to the testes. I love this picture. Is it this one? Where was it at? No, not that one. There we go. Notice this one, again, you don't need to know the origin and insertions of the abdominal muscles, but here we can see the aponeuroses of all of these muscles, and we can see how it's that inguinal ligament from the superior anterior iliac spine to the, no, this is the inguinal ligament. Remember the linea alba is that line on the midline, the white line on the midline. This here is the inguinal ligament. So this is the inguinal ligament. Here is the inguinal ligament. This is the linea alba. And then the fourth structure you're responsible for is this. Again, it looks like an aponeurosis, but it's really not. It's stabilizing multiple muscles. So this is actually a fascia in the thoracic and the lumbar region. So this is the thoracolumbar fascia. So those are the 4 structures you are responsible for in the trunk region. All right, questions on that? Great question. So the way let's go actually to our bones again. We're going to need to decrease that and go here. Excellent. So notice the latissimus dorsi has a massive origin. Let's go ahead and draw it. The origin of the latissimus dorsi basically is the lower 6 spinous processes. Oh, that's huge. I don't want that to be so big. Is that better? A little bit. So let's undo that, undo that. It is the 6 spinous processes of the lower 6 ribs. 1, 2, 3, 4, 5, 6. Here. The lumbar, the spinous processes are the lumbar vertebrae, right? And also the sacrum. Notice it is here on the crest of the ilium, a little bit from the inferior angle of the scapula, and some from the ribs. Basically, what happens here is this big, broad muscle has fascicles that basically come from all these locations to come around. This one comes this way, and these are coming this way, and this is all coming this way. We have all these that are coming off of the spine and all coming together to that single insertion. But we also have some, and I'll change the colors of them just to emphasize it, that are coming off of, and I guess some coming off the crest of the ilium and coming up this way, some coming off of the ribs and coming up this way, and some of them even come from the inferior angle of the scapula and come forward. So all these fascicles, this is one of those convergent muscles where you have this big fan-like orientation of the fascicles from all these parts where they're all coming together. And remember, they're all inserting, and I'm going to highlight this in instead of drawing it, because remember, it's on the anterior side, into that intertubercular sulcus on the anterior part of the arm, of the humerus. So they all come up underneath and onto the front of the arm. So really it's just this, and now I've drawn this horribly, but I've drawn it. But if we go back and look at the picture, we can see how it is this big, huge, broad, fan-like convergent muscle where all the fascicles, right, some coming off of the crest, some coming off of the ribs underneath here, some even coming off of the inferior angle of the scapula become part of this structure. Oh, great. That was another question too. Absolutely. So yes, absolutely. The modifications of the neuromuscular junction are on the test. They're part of the 7 possible essay questions that you can have. Yes, masseter would be the right answer. Brings it up. The other one you should have been torn between is the temporalis, because both of those are going to elevate. Notice the orbicularis oris doesn't close the mouth. It purses the lips. So that's the one there. So yeah, let's talk about the, let's talk about that modification. So, okay, let's switch gears. I think the whiteboard will be best for this. I don't remember if I took a picture from this. So let me do that again, just in case, because I thought that was cool. All right, and now we can go ahead and clear these. When we talk about, where's my text? There we go. All right, communication at the neuromuscular junction. As we talked about, there were basically 3 examples of toxins that we used to be able to see how we could modify it. There's certainly other ways we could modify it as well, but this was one of the ones, the 3 that we talked about in lecture. One of those, as was mentioned, was the Botox toxin, which again really is the botulinum toxin, the toxin of the botulinum bacteria. And as we talked about, when you're thinking about how it modifies it, basically there are 2 things to think about. The first is, what does it actually do? And the second thing to think about is the effect it's going to have on the muscle. All right, those are really the 2 things to think about. Now, remember with botulinum toxin, we said that is the one that breaks that protein. Again, it's called the SNAP protein. You don't have to know that, but we don't need to know that it breaks a protein in the synaptic end bulb. Right? So it breaks a protein in the synaptic end bulb. And the effect of the breaking of that is that now the synaptic end bulb, and because I wrote it out once, oh, what happened? Oh, I wrote it in the wrong place. Now the synaptic end bulb cannot release acetylcholine, right? Or more specifically, what it is that our vesicles cannot bind to the plasma membrane and cannot release the acetylcholine by exocytosis. All right, so the vesicles cannot bind to the plasma membrane. All right, that is what the botulinum toxin actually does. So then the next question becomes, what effect does that have on the muscle? And as was mentioned, as you had mentioned in the question, if I can't release acetylcholine from the synaptic end bulb, then I can't open those voltage-gated cation channels. If I can't open the chemically gated cation channels, if I can't open those chemically gated cation channels, sodium can't come in, I can't depolarize, I can't get a muscle action potential, right? And again, we can write that all out. If it was an essay question, I'd do that, but I'll do the short version here, right? There's no muscle, let's be even a little bit more. I'll give a little bit of it. No sodium enters into the muscle cell. And again, you would have to explain why. But the key of that is there's no depolarization. There's no muscle action potential. And if there's no muscle action potential, the muscle does not contract. All right? And as you mentioned in the question, we call that a flaccid paralysis. Because the muscle cannot contract. Even though I'm sending a signal trying to get the muscle to contract, because of that broken protein, it could not contract. All right? Now, we had another toxin that produced a flaccid paralysis, and that was curare. Oops, if I spell it right. Curare, and again, the same thing. What does it actually do? And as we mentioned, curare, which again is that toxin that is in like some poison plants, but mostly poison like mushrooms or poisonous frogs. They use it for making those blow dart poisons. What it actually does is it binds and blocks. There you go. Excellent. It binds and blocks. Specifically, it binds and blocks the chemically gated cation channels on the motor end plate. Right? It gums it up. Right? When we then think about the effect that it has on the muscle, well, if our cation channels cannot open, oops, I spell open right. They cannot open, again, no sodium into the cell, no sodium into the cell, no depolarization, no depolarization, no muscle action potential, no muscle action potential, no contraction. Again, we got that flaccid paralysis. Even though we were trying to control the muscle and getting the muscle to contract, it won't. The last toxin we talked about, though, was tetanus. Tetanus, again, you get from that rusty nail or that rusty piece of metal. And again, it is a toxin that gets in from a bacteria associated with that. And again, we want to know, what does it actually do? Tetanus is similar to curare in that it binds to the chemically gated cation channels on the motor end plate. However, here is where it's different. Whereas curare binds and blocks, tetanus binds and activates. Right? That channel. Basically, it opens the channel. And remember, it doesn't let go. So it doesn't let go. All right? So when we think about the effect on the muscle, well, now it has cation channels that are constantly open. Exactly. And since it's constantly open, sodium is constantly coming in. Right? We are continuously depolarized. So we are constantly producing muscle action potentials. And if we are constantly producing those muscle action potentials, the muscle is in a constant state of contraction. The muscle contracts and won't stop contracting. And again, we still can't control it, so it is a paralysis. But this time, it is paralyzed because it won't stop contracting. And so, as we talked about, we called that a rigid paralysis. All right. So again, the muscle is stiff, the muscle doesn't. All right. So there you go. So those are the three examples of the toxins that we used to modify communication at the neuromuscular junction. And absolutely, I could ask about one or more of those on the exam. All righty. Any more questions?
I do, but I hate typing. So, what is the answer on the muscle activity? The throw a ball up in the for a tennis serve, and then the bowling ball. I was kind of confused on the both of those.
So if you think of in terms of a tennis serve, right? What you have to do for a tennis serve is you have to have the racket up to be able. I think I actually used this example in lecture, which is why I did this. You have to have the racket up. So to do that, you have to elevate your shoulder, you have to bring the shoulder back, and you have to rotate the shoulder to get the tennis racket up to be ready to hit it. Then to serve the ball, what do you have to do? You have to throw it up in the air. And if you're going to throw it up in the air and you're going to hit it, you have to extend your head to be able to see it. So what muscle elevates, retracts, and rotates the scapula and extends the head? Oh, so it looks like someone already gave you the answer.
And would the ball be the brachialis?
No, look at the chat.
Oh, yes, for the
Trapezius.
tennis one, right?
Right, 4 actions of the trapezius, absolutely. Right.
For
If you're going to throw a bowling ball, you have to flex the elbow, pardon me, flex the shoulder, flex the elbow, and to hold the ball, you have to be supinated. So what flexes the shoulder, flexes the elbow, and supinates the hand? Someone help her out.
Is it the brachioradius?
Yeah, bicep brachia.
Okay.
Right. So again, think of the actions, the 3 actions, and that's what. And again, I just wanted, and the other thing you had to do was that in reverse. Think of the 3 actions and put them together into some type of activity that would allow you to use it. Again, normally this is just a fun activity that we do in the class as a way to get mnemonics to help us to study this, but getting you to do this exercise, and that's really kind of what we're doing online. We're finding ways to force you to think about the anatomy of this so that we get the lab component. So that's why this, instead of just being a fun activity that I stand up and I dance around like a monkey and we all laugh and have a good time, and then you stand up and dance around like a monkey and we laugh and we all have a good time, becomes a great assignment to make sure that you guys are thinking about the lab portion of this class. I do not drop any scores. Again, every test matters. However, I do reward you for gaining this material and retaining this material. So remember, I don't drop a score, but what I do allow you to do is replace a score with your final. because we have that weird third exam, I don't know if I'll be doing it exactly or percentage-wise or how exactly that will work, but they're absolutely, while I have not figured out what it is going to be yet, because like I said, you had your lab and lecture exam on Thursday. My 431 has their lab and lecture exam on Wednesday. So I literally spent my entire break working on writing those 4 exams. And so I haven't thought that far in advance. I guarantee you there will still be some reward for being successful on the final exam that can help the student who has learned how to be successful moving forward to do that. I don't know if we'll be able to straight replace an exam anymore, but I guarantee there will be definitely some benefit. Because again, the point of this, I know for you as students, is you want to get your A or whatever grade you need to move on to whatever program you want to do. But my goal in teaching you 430 is not only to get you to successfully know this, but most importantly, give you the information that's going to help you to be successful in 431. None of the stuff that I've given you has just been randomly pulled out of the hat, right? The bone features are things we're using for the origins and insertions now, or they're going to be the holes that nerves pass through, or they're going to be the holes that blood vessels pass through when we get to 431, right? Everything, there's a reason why I'm teaching you everything that I'm teaching you, and it's either to help you to be successful in this class or in 431. So again, you need to gain and retain this information. And so by showing me that you can do that on the final, I will definitely reward you. I just don't know exactly. My guess is it still will be some type of replacement. I have to look at the math and see how we'll do it with our third exam because it was worth so much less. All right, because I don't want someone who's because it was the first time we did something online, was their lowest score, that they'd be penalized somehow. So I'm not sure how the math will work out on that, but I guarantee there will be some reward for being successful on the final. I just need to see where we're at the end to see exactly what that will be. Well, as I said, I will probably get the exam posted tonight or maybe tomorrow, depending on how things go. I'm close. Like I said, I need to fine tune it. And then there's also a couple tweaks that I need to do and make sure that there aren't any problems with it. But once I get that done, I'll get it posted. But again, remember, it's going to be locked till 10 o'clock. And I've already told you how many questions are going to be on it, so you know what to expect from all of that. But it will become available 10 o'clock on Thursday. Like I said, and that's the one last thing I want to point out. Remember, there are 80 questions. Oops, I didn't mean to do that. That's fine. I don't care. Get that out of the way. Let's do that, actually. Oh. Right? If you think about it, the lab exam is 90 minutes. Right? The lecture exam, you have 150 minutes. Right? That is 4 hours to take these exams. Right? You should take a break in between. So make sure you're giving yourself enough time. Now, it probably shouldn't take you that amount of time, but I know some people are a little bit slower. If you need that full time, take it, but then make sure you plan accordingly. Remember, the exams end at 6. So if you need 4 hours and a half an hour break in between, don't start the exams any later than 1:30, right? Probably start them by noon or something like that, just to make sure that you have time if there's any problems or anything like that. Yes, you will be. So to answer your question, with the proctoring, you will be recorded, and I will be viewing them. So I will be viewing your recordings. So yes, so again, the way the proctoria is set up is that it is monitoring your activity, and anytime there's suspicious activity, that suspicious activity is flagged. And so I have the opportunity to go. And again, it's what it calls suspicious activity. Suspicious activity could be someone closing a door. So there's a loud noise, a noise that is, or you coughing or sneezing could be something that is considered. But remember, it's also watching your eye movements as well, which again, so you shouldn't be looking down at notes or your phone or anything like that. You shouldn't be getting up, moving around. If there are problems with your scanning of your area, if you're not scanning your work area, all of those are things that will be flagged, And so then I get to have, not only do I get to grade your exams, but I get the joy of watching these Proctorio recordings to observe the activities that it believes are suspicious, to see if it actually is suspicious activity, in which case we will have a discussion. But in most cases, it is benign and there are no effects. The Proctorio itself does not penalize you. So if your kid runs into the room screaming, you're not going to get instantly kicked out of the exam, right? What'll happen is there'll be a flag. I will observe it and see that it was just your kid running in and screaming, and it's not going to be a big deal. All right, so don't have to worry about getting, being kicked out or anything like that. But you will be recorded, and I will be observing it, because the same way I look around during the test to make sure you're not cheating, this is unfortunately what has to be done to make sure that students aren't cheating during these types of exams. So yes, I get the joy of getting to watch you guys. So wear funny shirts or something, I don't know, funny hat or something, right, to amuse me while I'm observing you during the exam. All right, but I will not be observing you during the exam. This is something that is recorded and I view after the fact. All right, so I can't peek at you while you're taking the exam. That's not how this works. All right. If you have more questions. True, I guess that there are proctoring services where they do have proctors who observe you and basically do the same thing. This is an automated version of that. I guess the one thing that I would say is the one advantage of having some random person doing it is they are, what's the right word? The automated one is going to be more conservative. There's probably going to be a lot more flags where there's a lot of things. Like I said, if you sneeze, it's going to flag. Where if some random dude is watching you and you sneeze, they're not going to flag that because they know you just sneezed, right? So if anything, a random person would have been less work for me, but it is what it is. And like I said, the good news is I will have had a real lab and lecture exam under my belt when we come into Thursday. So there may be some subtle changes, or maybe not subtle changes, that occur Thursday morning before the exam based on what happens Wednesday morning. Wednesday, their exam starts at 7 because that's my morning class. So they're done from 7 to, I think their class goes till noon or 2 or something like that is their range, whatever that is. So I will have, a good, 16, 18 hours if something needs to be adjusted, which hopefully won't be the case. Hopefully this will be made properly, and I'll get it beta tested and make sure that it's working. So like I said, it'll probably be posted. My guess is tonight because I want to be ready for tomorrow's exam. So you'll be able to see that the test has been published, but like I said, you won't be able to access it till Thursday. All right, I think at this point, unless there's any more questions, what I'll do is I'll go ahead and stop the recording. Like always, I'll hang around a little bit if anybody has any remaining questions or anything like that. You guys have been great about emailing questions, so by all means, if you have questions, email. In fact, ironically, all during spring break, nobody came to my office hours when I was in Zoom, but I got a ton of emails, including several students who actually emailed me during my Zoom office hours. So rather than coming in and texting and talking to me, they sent emails instead. But if that's how you're more comfortable communicating, that is fine. But I can meet you on Zoom if you have questions. We can still do that. Or email me questions. And obviously, with my 431 and your exams coming up, I will be constantly monitoring my emails. It also keeps me away from my family, which is nice. So feel free to contact me if you have any questions, and I'll see what I can do to help you. All right. I wish you guys tremendous success. Study hard. Like I said, I honestly have no idea what this test is going to be like. I think literally every question that I have ever asked on any exam ever, plus a bunch more that I made up, have been added to this. So we'll have to see how it goes up. I have no idea what this is going to be like. It'll be an interesting experience. But the good news is it's all stuff. Again, you've had 4 tests now. You know how I ask the questions. You know what I want from the information. There's nothing new on this. This is all stuff that you've seen before, and it's all off your list, and it's all off your lectures, and it's all off the things that we've talked about. So hopefully, as you've been learning to be successful at this, you've been learning the things that will help you to be successful. And just do your best, be descriptive, right? And again, pay attention to your time. When you reach the end of that time limit, you get kicked out of the exam. So make sure you're paying attention to your time. And like I said, if you need it there for the lecture exam, there is that that whiteboard tool on there. Do not have your phone out for any reason. Do not have any papers or notes or books or anything like that out. None of those things should be out and around during the exam. All right. Good luck. Have fun. And I will talk to you soon. And then, that's the last thing. After the exams, because we'll have 5 days before we come back in the nervous system, I do have a nervous system activity that I'm going to have you do, so you can start thinking of that, because the nervous system has less anatomy than the muscular system, but it's less intuitive, right? You can look at a muscle and see its origin and insertion and be able to guess at what its actions are. You can't, the part of your brain that moves your hand doesn't look like your hand, right? The part of your brain that allows you to see doesn't look like an eyeball. So it's less anatomy, but I also think it's less intuitive anatomy. So, and a lot more physiology, a lot more physiology. So I will, again, after you take the exam, take a good 15, 20-minute break, and then I want you diving right into the next material. I don't know if it's harder because while it is less intuitive, it's still less information. So again, some people, because there's less, they do better with it because it's less intuitive. Other people's do worse. All I can tell you is statistically. Statistically, the scores are similar. I would say, like I said, both the muscular system and nervous system exam scores are similar. So I guess that means similar hardness. Bones are typically one of the easier ones, and so typically the grades are higher on that. And the tissues is typically the one that is the lowest. So all I can say is mathematically, based on the scores, they're similar hardness. It's just a different type. It's a different section. We're going to study it a different way. And so in some ways it'll lend itself better, I think, to being online, because it's you don't hold the brain in your hand the same way that you hold an arm and leg in your hand and look at the muscle and see how it works. So I think that could benefit us. But that's a problem for another day. We don't want to worry about that till after Thursday. All right. All right, I think that is good. I'm going to go ahead and stop the recording now. Again, I wish you guys luck. If you have any questions between now and then, please let me know. All right, take care. I'm going to stop recording. Like I said, I'll hang out for a little bit if anybody has any questions. Otherwise, good luck on the exam.